Hi everyone, welcome to Medical Microbiology. Today we will discuss Chapter 4, Survey of Prokaryotic Microorganisms. On the first lecture, we're going to learn a little bit about living things. So all living things will have these 5 characteristics. And these characteristics distinguish between living things and non-living things. Living things will have this characteristic, the non-living things will not have this characteristic. The first characteristic is all living things grow and develop. Okay, so growing means is become bigger or taller by adding more cells. Development means these living things, so some of them, like for example humans, cells that growing, they also differentiate into different organs. There are cells that become liver, the heart, the lungs, and etc. So this is development. The second characteristic of life is movement and irritability. So all living things has the ability to respond to the stimulus. They have the ability to move, whether a whole body movement, like human move from one place to another place, or movement inside the body, like for example, movement of water inside the plants, okay, from the root to the leaf. The third characteristic of life is all living things has the ability to support themselves. Like for example, they have the ability to absorb the food, find the food, eat the food, okay? They have the ability to transport materials, chemicals, molecules from outside to inside or from inside to outside the cell. They also have the ability to protect themselves by producing enzymes, antibodies, and etc. So these are the characteristics of life, the third characteristic of life. The fourth characteristic of life is metabolism. What is metabolism? Metabolism is all chemical reaction that present in the cell or in the body. There are two types of metabolism. Okay? The first one is called anabolism. Okay, anabolism means synthesis reaction. So this is the reaction that builds bigger molecule from smaller molecule. And the second metabolisms will be catabolism. Okay. This is the chemical reaction that break the bigger molecule into smaller molecule, or this is also known as decomposition. So all living things will have this ability to perform metabolism. And the fifth or the last characteristic will be reproduction and heredity. So all living things have the ability to reproduce because with their reproduction, they will stay in this world. So make them become exist. If they are unable to reproduce, then they undergo extinction. So reproduction means production of new individual, okay? And heredity is related to the ability to transfer the gene, the characteristic from parents to the offspring. So these are the 5 characteristics of life. All life, okay, or living thing, they consist of cell. Right? Consists of cell. So cell is actually the smaller unit of living. All living thing will have cell. There are some organisms that only have one cell, okay, single cell organism, or sometimes is also known as unicellular organism. The organism that only have one cell and they are able to perform all the five characteristics of life. Example is all bacteria will be single cell, and there are some organism is called protozoa, algae. They're also single cell organism, okay, or unicellular organism. The second type of organism is called the multicellular organism, the organism that has a lot of cells in their body. Like human, for example, human is multicellular organism because human have trillion of cells. Okay, so this is the second type of organism, multicellular organism. Now all cells have the shape, okay, how it look like. Basically, there are three basic shape of cell. The first one is look like a ball, round. It's called a spherical. The second one is look like a cube. Okay, it's a cubicle. And the last one is look like a cylinder. It's long cell. Okay, this is cylindrical. Okay, so these are the three basic shape. But there are other shape, like there are some cell that look like a spiral. Okay, so there are some cell that look like a crescent moon. Okay? But again, there are three basic shape of cell: okay? Round, cubical, or cylindrical. All these cells, okay, will have some similarities. The first one will be, okay, all cell will have the boundary. Okay, that boundary is called the cell membrane. So all cell will have the cell membrane. The second, okay, all cell will have the cell contents, which is called the cytoplasm. So this is cytoplasm. This is a cytoplasm. Okay, the content of the cell is called the cytoplasm that mostly consists of water. And then all cell will have the chromosome, okay, because all cells have the ability to reproduce and to transfer the gene. So this gene is actually located in the chromosome. So therefore, all cells will have the chromosome. Okay, this is chromosome. And all cell will have the small organelle that able to produce proteins. That organelle is called ribosomes. So all cells will have ribosome. So there are again four structures that will be present in all cells, which is the first one is cell membrane, the second one is the cytoplasm, and the third one is the chromosome, and the fourth one is the ribosome. So all of these four structures will be present in all types of cells. There are two main basic types of cell. The first one is called the eukaryotic cell. The second one is called the prokaryotic cell. Eukaryotic cell mean this cell has nucleus and other organelles inside the cell. Eukaryotic cell actually mean, okay, nucleus. They have real nucleus. Eu mean real. Okay, karyon mean nucleus. Okay, so from the name, then we know that eukaryotic cell means the cell that have real nucleus. Prokaryotic cell, okay, pro mean before, okay, pro mean before, and karyon mean nucleus. So it means that this cell do not have nucleus, okay, do not have nucleus. Now, what is nucleus? Nucleus is actually the chromosome plus the envelope that protect the chromosome. So when the chromosome is protected by this structure, the envelope, then it's become nucleus. Okay, so eukaryotic cell will have this nucleus, while the prokaryotic cell will not have the nucleus. Okay, now we're gonna compare these two types of cell, prokaryotic cell and eukaryotic cell. Now, just for information, for easy remember that only one organism that have prokaryotic type of cell, which is bacteria. Okay, just remember that all bacteria, they are prokaryotic cell. The rest of organism will be having the eukaryotic type of cell. So like protists, fungi, plant, and animal. Okay, all of them, all of these organisms will have eukaryotic type of cells. So just for easy memorization, all prokaryotic cell will be bacteria, and eukaryotic cell will be all other organisms. Okay, protists, fungi, plants, and animals. Now, we're going to see the similarities first. Okay, all cells, remember, they will have the cell membrane. So it doesn't matter whether prokaryotic or eukaryotic cell will have the cell membrane. Similar, the same cell membrane. Then the second structure will be cytoplasm. So all cell will have cytoplasm, the cell contents. And all cell will carry chromosome because all cells have the ability to reproduce and transfer their gene. So therefore they will have the chromosome. And all cell will have the ribosome, which is the organelle that used for making proteins. So these are the four similarities and the four structures that present in prokaryotic cell and eukaryotic cell. Now the differences will be the eukaryotic cell, their chromosome will be protected by the nuclear envelope. So this is nuclear envelope. Protect the chromosome, and this whole organ, this whole structure become nucleus. So nucleus is actually chromosome that is protected by the envelope. So this is the nucleus now. And this is only present in the eukaryotic cell. And eukaryotic cell also have other organelles. What is organelle? Organelle is small organ inside the cell that has special function. So like for example here, eukaryotic cell will have rough ER, rough endoplasmic reticulum. And this rough ER has a function to store the proteins. Okay. Remember ribosome, they produce the protein and it will be stored in the RER. And the next organelles in the eukaryotic cell is smooth ER. The function of smooth ER is to produce proteins and lipids. Okay. And then there is another bigger organelle over here. It's called the Golgi apparatus. The function of Golgi apparatus is to modify lipids, proteins, and carbohydrates into final products like enzymes and hormones. So Golgi apparatus actually will take the protein from RER, the lipid and carbohydrate from the SER, and then it will modify that molecules into a new product. So basically, Golgi apparatus is the factory inside the cell that produce enzymes, hormones, okay, and other products. There is another or other smaller organelles like lysosome. Lysosome carry enzyme. It's called a lysozyme that used for destroying bacteria. Maybe there is bacteria that enter into eukaryotic cell. It will be destroyed by this lysosome. There is another organelle. It's called peroxisome. It's carry enzyme that destroy the toxicant or drugs. Okay, so this is for detoxification. And there is other smaller organelle, it's called the proteasome. This is the organelle that will destroy the extra unnecessary proteins. Okay, so like for example, maybe ribosome produce some unnecessary proteins will be removed or destroyed by this proteasome. And there is mitochondria, which is the organelle that produce the energy, ATP, for all the process inside the cell. Okay, mitochondria is the power, okay, the power plant inside the cell. There is also organelles like chloroplast that only present in plant, okay, and algae. And this organelle is used for making carbohydrate, the food, okay, glucoses. So these are the differences between prokaryotic cell and eukaryotic cell. Okay, so again, there are two types of cell, eukaryotic cell, including animal, plant, fungi, and protists. And protists consists of protozoa, okay, which is a primitive organism that look like animal, and algae, which is a primitive organism that look like plant. Okay, so that's protist. And prokaryotic cell, of course, all bacteria. Bacteria are prokaryotic organism. Okay, so remember in chapter 4, we will learn about the prokaryotic cell structure. Okay, so there are three main structures. The first one is called the external structure, which is the structure that located outside the cell and usually used for maybe protection, attachment, movement, and etc. The second one is called the cell envelope, which is the boundary of the cell. For the prokaryotic cell, remember bacteria. Okay, so prokaryotic cell will be bacteria. They have two membranes, which is the cell wall and the cell membrane. And the last one will be internal structure, the structure inside the cell, of course, cytoplasm, chromosome, and ribosome. And there is other structure like plasmid that only present in bacteria, inclusions and granules, cytoskeleton, and some bacteria has the ability to produce the endospore, which is the structure that is really difficult to destroy, to kill. Okay? So these are the structure of prokaryotic cell. We will learn this on the second lecture. Okay, so we finish our first lecture, then we will meet again on the second lecture, which is basic structure of the prokaryotic cell. Goodbye now. Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 4. This is lecture 2, the structure of bacteria or prokaryotic cell. On lecture 2, we will discuss the external structure. Okay, this is the summary for the structure of prokaryotic cell. Okay, remember it has three main parts: the external structure, the cell envelope, and the internal structure. So today in lecture 2, we just focus on the first part, which is the external structure. Okay, the external structure, which is the structure that's located outside the cell. Okay, some of them has a function as attachment, motility structure, and some of them has the function as protection. Okay. So the first type of external structure is what we call the appendages. Appendages mean the structure that can be used for movement, attachment, and some transfers of materials. There are two major groups of these appendages. The first one, of course, the appendages that are used for movement. This is what we call the motility appendages. Example is flagella and axial filament. Okay. So flagella usually presents on the bacteria that have rod or bacillus shape. So they usually have flagella for movement. While the axial filament is usually the motility structure or movement structure that located on bacteria that has spiral shape. Okay, so this axial filament will be located here, which is this spiral type of bacteria will move like glide, gliding movement. The next type of appendages is appendages that used for attachment and also used for channel, okay, for transferring some chemicals, especially transferring the genetic materials. The one that used for attachment is called the fimbriae. Okay, it's gonna be look like this. It look like hair structure around the bacterial cell. And the one that used as a channel is called pili or pilus if it is only one. So like, for example, this is one bacteria, there is another bacteria. One of them can transfer the genetic material using this pili. It looks like bridge between two cells. The second type of external structure is called the glycocalyx, which is the materials that cover the whole cell. Okay, it's a surface coating of the cell, usually used for protection. We start with the first type of appendages, which is used for motility. Okay, so it's for movement, which is the flagella. Flagella of bacteria, it has three parts. The first one is called the filament, which is consists of protein. So this is the first part over here, the filament, which is long proteins that go, from inside the cell to outside the cell. Okay, so it look like kind of tail. Okay, so this is the filament. And then the one that attached to the cell, bacterial cell, is the hook. So the hook gives the strength for the filament, for the tail. Right? And the last one, okay, will be the what we call the basal body. Okay, so this is the basal body, which is the one that anchor the whole flagella inside the cell. Okay, so this is the basal body. Now the movement, okay, how the bacteria move. This is really important because there are some of the eukaryotic cell like protozoa, they have also flagella. The way they move gonna be different. Okay, it will be different. The prokaryotic cell move by rotating the flagella, so make a rotation. Okay. The eukaryotic cell, like protozoa, for example, they move by whipping the flagella, okay, their tail. So these are principal difference between the movement of prokaryotic cell or bacteria and the eukaryotic cell. Prokaryotic cell or bacteria move by rotating their flagella. The eukaryotic cell, like protozoa, move by whipping their flagella. There are several types of bacteria, okay, based on the flagella. The first one is called monotrichous. Mono means one. Trichous means flagella. Okay, it means that this bacteria only have one flagella. So monotrichous. So one flagella. It's monotrichous. The second one is called lophotrichous. Lopho means bundle. Okay, so this bacteria basically has bundle of flagella in one side. So a lot of flagella, but only in one side of the cell. That's the lophotrichous. The next one is called amphitrichous. Amphi means both sides. It means that this bacteria has flagella on both sides, on the basically on the opposite side. Okay, so that's the amphitrichous bacteria. And then the last one is called peritrichous. Peri means surrounding. Okay, so this is the bacteria that has flagella that's located on around the cell. Okay, so around the cell. So it has a lot of flagella around the cell. That's very tricky type of bacteria. Okay, now again for the movement, flagella, the bacteria that have flagella will move by rotating the flagella. The way they move, if it is counterclockwise, which is to the left, okay, then this bacteria will move forward or run forward. Okay, so that's the way they move, by counterclockwise rotation of their flagella. If it is clockwise, which is a right turn, okay, turning to the right, okay, rotating to the right, then this bacteria will be stopped or tumbles. Okay, so they will stop or tumbling when they rotate their flagella clockwise. They move based on stimulus. Okay, like for example, food. Yeah, food is a stimulus. It is chemical. Okay, so bacteria will move toward the food. Okay, this is what we call the chemotaxis. Chemo mean chemical, taxis mean movement. So they move to the chemical stimulus. It might be positive or negative. If it is food, then it's going to be positive chemotaxis. But if it is chemical, like a poison, for example, eh, poison is also chemical, then the movement will be chemotaxis negative because basically the bacteria will run away from the poison chemicals. Okay. If the stimulus is the light, then the movement is called phototaxis. Again, it's going to be whether positive or negative. Okay, so positive phototaxis means the bacteria will move toward the light. But if it is phototaxis negative, the bacteria will run away from the light. Okay, so this is again how bacteria move by rotating their flagella. The second type of flagella is called the axial filament or periplasmic flagella. This is usually present on bacteria that has spiral shape. So this is flagella around the cell, okay, following the spiral shape of the cell. And when these flagella move, okay, the axial filament move, then it will give gliding movement. So axial filament is used for bacteria to glide. Okay, so this is especially for bacteria that have spiral shape. Now we go to the next type of appendages, which is called the fimbriae. This one is not for movement. This is the function is for attachment. Okay, so bacteria that have fimbriae, they able to attach in the wall of their host cell. Like for example, E. coli, they do have the fimbriae that make them able to attach to our intestine. Okay, so E. coli, they have the fimbriae for attachment. So this is a very fine protein structure that look like hair. Okay, look like a bristle of hair. So again, the function is for attachment to the host cell. The next appendages is pili. Okay, this one is called pilus or pilus. Okay, so this appendages is used for transferring materials from one bacteria to another bacteria. Usually the one that is transferred is DNA or genetic material. Okay, like for example, resistant genes. Okay, so there are some bacteria that really difficult to destroy. So because what? Because they have resistant genes, they can transfer this resistant gene to other bacteria. Okay, to other bacteria. So the one that transfer the gene is called positive bacteria. Gonna change the color over here. So the positive bacteria, this is the donor. Okay, and then the one that receive the gene is called negative bacteria or recipient. Okay, so this is donor, this is recipient. This one may be also recipient. Okay, so they can transfer from to more than one bacteria. So this is pili. So if you see this structure, this over here, this is like a channel that used for transferring this gene. That's the pili. Okay, so again, pili is the structure that is used for transferring material, like gene, for example. The next appendage is, I'm sorry, the next external structure is glycocalyx. And this glycocalyx is mostly used for protection. There are two types. Yeah, the first one is called slime layer. It is like a gel. Okay, it is not really solid. Therefore, it's called slime layer. Usually, the slime layer will be produced to attract other bacteria to come. Like, for example, in the mouth, around the gums, there are some bacteria that live in our mouth, produce the slime layer. And this slime layer will attract other bacteria to stay in our gum. And then the second type of glycocalyx is called capsule. And this is really solid. Okay, it's a solid glycocalyx, mostly used for protection. Okay, it will protect the bacteria against, for example, maybe the host antibodies. So they, protect themselves from the antibodies of the host cell. I think that's enough for this first part of the bacterial cell structure, which is the external structure. Then we will meet again on the next structure, the second type of structure, which is the bacterial cell envelope, on lecture number 3. Goodbye now.
Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 4, Structure of Bacterial or Prokaryotic Cells. On lecture 3, we will discuss about cell envelope, which is the cell wall and the cell membrane. Let's see the summary of the structure of the prokaryotic cell. Remember, it has 3 main structure. The first one is what we call the external structure. Okay, so external structure that consists of appendages and glycocalyx. The appendages mostly used for movement like flagella and axial filament, or maybe used for attachment like fimbriae, or used for transferring the genetic materials like pili. The glycocalyx, it's used for protection and also to produce the biofilm, which is the attraction for other bacteria to come and to make the community of microorganisms. The second structure, okay, the second group of structure is called the cell envelope. This is the one that we will study today. Okay, the cell envelope of bacteria or prokaryotic cell consists of 2 main layers. The first one is called the cell wall, and the second one is the cell membrane. Okay, again, the cell envelope. The cell envelope consists of cell wall and the cell membrane. The primary function of the cell envelope is for covering, protecting the inside structure of the cell. It is also used for maintaining the cell integrity. It's also used for movement of materials in and out. There are two different groups of bacteria, okay, based on the cell wall. And this is the two main group of classification of bacteria. The first one is called the gram-positive bacteria. Okay, it has a very thick cell wall. And the second group is called the gram-negative bacteria. It has a thinner cell wall. Okay, so that the two main group of bacteria based on their cell wall. Now the cell wall, the structure again mainly, okay, the main molecules that make up the cell wall is called peptidoglycan. So this is the main molecules that make the cell wall of bacteria. Remember, algae, plants, fungi, they also have the cell wall. But the molecules that make their cell wall will be different compared to the molecule that make the cell walls of bacteria. So the cell wall of bacteria made of peptidoglycan molecule. And from the name, we should be able to guess, okay, that this molecule actually consists of peptide or peptide, which is protein or amino acid, and the glycan, which is glucose or carbohydrate. So this molecule actually composed of amino acid and carbohydrate. Okay, this is a lot of amino acid over here. This is all the amino acid, and this is the carbohydrate. So this carbohydrate connected with this peptide or the amino acid. There are two types of bacteria based on the cell wall. The first one is called the gram-positive bacteria. Okay, so gram-positive bacteria, which is, look at this cell wall over here. The cell wall is really thick. So the peptidoglycan, okay, the peptidoglycan wall is very thick. So this is gram-positive bacteria. And then under that cell wall, of course, this is the cell membrane over here. Okay, so this is cell membrane, and this is cell wall. So that's the gram-positive bacteria. The gram-negative bacteria, if you see the cell wall, is very thin, and this cell wall is actually located between the cell membrane and other protein layer on the top. Okay, so this is the cell wall. This is the cell wall, which is the peptidoglycan over here, which is really thin, okay, compared to the gram-positive bacteria. And of course, this is the cell membrane. Okay, cell membrane. At the top, there is another layer of proteins, okay, which is outside, okay, outside the cell wall. So basically, this gram-negative bacteria has 3 layers on their cell envelope. Okay, the protein layer, the outer membrane, okay, and then the cell wall and the cell membrane. Okay, so 3 layers over here. 1, 2, and 3. Now, if we compare side by side, again, you see that this gram-positive bacteria only have 2 layers, okay, of the cell envelope. The first one is this one, which is cell wall. And this second one is this, which is cell membrane. Okay, and this is the gram-negative bacteria. It has 3 layers. One, this is the outer membrane. Outer membrane. This one is the cell wall, which is really thin. And number 3 is the cell membrane. Okay. So these are difference between the cell wall of gram-positive bacteria and gram-negative bacteria. So during the lab experiment, you will do the experiment, which is staining the bacterial cell, and then from the stain procedure, the gram staining, you will be able to know which one is gram-positive and which one is gram-negative. The gram-positive bacteria will have blue or purple color under the microscope. The gram-negative bacteria will have red or pink color under the microscope. This is another comparisons in the table. Okay, again, the number of, this is cell wall. Okay, so cell wall. The number of layer of gram-positive only 1, which is really thick, peptidoglycan. This one has 2, okay, which is outer membrane, and thin peptidoglycan. Okay, so this one is thick, oh sorry, peptidoglycan. Okay. Again, chemical composition of the cell wall, the main molecule is called the peptidoglycan, and there are some other molecules that also present on the cell wall. For the gram-negative bacteria, okay, of course, peptidoglycan, but it's really thin. And above that, there is a outer membrane that consists of lipoprotein, lipopolysaccharide layer. And then the thickness is really thick, between 20 to 80 nanometer. And gram-negative bacteria is only between 8 to 11 nanometer. Outer membrane, no. Okay, there is no outer membrane. This one, yes, it has the outer membrane, which is consists of lipoprotein and lipopolysaccharide. Periplasmic space is very narrow, and this one is very wide or extensive. Permeable to molecule is more penetrable, and this is less penetrable. Okay, this is also give us the idea that the gram-negative bacteria will be more difficult to destroy by antibiotics. Okay, because they are less penetrable. This one is more penetrable, mean it's gonna be easier to destroy by the antibiotic. Now, the gram stain which is the staining procedure to identify whether bacteria gram positive or gram negative. Okay, so again, gram positive will stain purple. The gram negative will stain red. Again, you will do this in the lab experiment. So just follow the procedure, the instruction during the lab experiment. And finally, if you do it correctly, you will be able to know or to identify which one is gram positive bacteria and which one is gram negative bacteria. Okay, that's the two main type of bacteria based on the cell wall, which is gram positive and gram negative bacteria. But there are some exceptions. There are some other bacteria which is actually really not many of them do not have the peptidoglycan. Okay, so they have different type of molecules that make up their cell wall. Okay, for example, is the TB bacteria. Okay, tuberculosis bacteria. They do not have the peptidoglycan. Okay. Their cell wall is actually consists of lipid mycolic acid, okay, which is gonna be difficult to stain with the gram staining. It means that they cannot be stained with the gram staining. You will not be able to tell whether they are positive or negative. Therefore, they have special staining procedure, which is called the acid phase staining. Okay, so the acid phase staining is especially used for this type of bacteria. Like for example, to identify the TB bacteria, then we use the acid phase staining procedure. You will also do this in the lab experiment. So just follow the instruction, follow the procedure, then you should be able to see whether the bacteria is TB bacteria or other bacteria. And there are also some bacteria that actually do not have the cell wall, and this is very rare. It's only group is called the mycoplasma. Okay, so mycoplasma is a group of bacteria that do not have the cell wall. Okay, no cell wall in this group, and this is the very rare type of bacteria. So mostly bacteria have cell wall except the mycoplasma. Now under the cell wall, okay, there will be a second layer is called the cell membrane. Remember when we talk about the differences or the similarities between prokaryotic cell and eukaryotic cell? All cell, whether this cell is prokaryotic cell or eukaryotic cell, they all will have the cell membrane. Okay, so all cell have cell membrane. Okay, but not all cell have the cell wall. Okay, cell wall only present in bacteria, plant, algae, fungi. But protozoa, animal, they do not have cell wall. But all of them, okay, all of them have cell membrane. So cell membrane is the layer that present in all type of cell. And they're all the same type of molecule that make the cell membrane is called the phospholipid. Okay, it's called the phospholipid, which is the molecule that has phosphate and lipid. Okay, fat. Okay, and they make two layer. Therefore, it's called the bilayer. So this is two layer of phospholipid molecule. If you see this one, this is one phospholipid molecule like this. It has head, okay, and then two tails. This is one phospholipid molecule. So they make layer. So one layer on the top like this. This is one layer. And there is another layer at the bottom. Okay. So it has two layers. This is what we call the bilayer. Okay, two layer of phospholipid molecule. So you see this one? This is one layer. Okay, and this is another layer of phospholipid molecules. So therefore, the cell membrane is also known as a phospholipid bilayer because it has two layers of phospholipid molecule. In addition of this main structure, which is phospholipid molecules, the cell membrane also have other molecule like proteins. So this one, this is a protein. This is another proteins. This is proteins. This is other proteins. This is some carbohydrates over here. Okay. So and it is okay. This one over here is cholesterol. Okay, so the main structure, the main framework is phospholipid bilayer, but there are some other molecules like proteins, carbohydrate, and also cholesterol present in the cell membrane. And the primary function of the cell membrane, of course, for protection and also for controlling movement of molecule, okay, from inside to outside or from outside to inside. Okay, I think that's enough for the cell envelope of bacterial cell, which is the cell wall and the cell membrane. Just remember, the cell wall of bacteria consists of peptidoglycan molecules. The cell membrane consists of phospholipid bilayer. We will see you again on the next lecture, which is internal structure of prokaryotic cell. Goodbye now. Hi everyone, welcome to medical microbiology. So today we will continue chapter 4, and this is lecture 4 of chapter 4. This is going to be the last lecture for chapter 4. Structure of bacterial or prokaryotic cell. And today we will study the internal structure, okay, of the bacterial cell. Again, this is a review of the structure of the prokaryotic cell. It consists of external structure, which is appendages and glycocalyx. We already learned this in lecture number 2. And then the cell envelope, right, which is consists of cell wall and the cell membrane. Remember, based on the cell wall, there are two types of bacteria, gram-positive and gram-negative. Right? We already learned this in lecture number 3. Now today, in lecture number 4, we will learn about internal structure of prokaryotic cell, which is cytoplasm, chromosome, plasmid, okay, ribosome, inclusion and granule, cytoskeleton, and endospores. Okay, remember from this internal structure, the one that have similarity with the eukaryotic cells are the cytoplasm. So all cells will have cytoplasm. And then chromosomes, okay, all cells will have chromosome and ribosome over here. Okay. So all cells will have this cytoplasm, chromosome, ribosome. Plasmid is very special in bacterial cell, including inclusion and granules. They are also present in bacterial cell. Cytoskeleton is actually also present in eukaryotic cell. And this one is really special because not all bacteria have this endospore. Only certain bacteria like Bacillus bacteria and Clostridium bacteria have the endospore. So this is a special structure of some type of bacteria. Okay, we start with the cell cytoplasm. So cytoplasm is actually the inside or the contents of the cell, which is majority is actually water. So 70 to 80% of cytoplasm is actually liquid or water. And there are some other molecules like a sugar, amino acid, salt, will be also present in the cytoplasm. But again, the main molecules is water. And then chromosomes. A chromosome is actually the one that carry the genetic material, which is the DNA. So the chromosome of bacteria is single, okay, kind of circular chromosome with double helix DNA. So yes, remember DNA is look like this. It has double stranded, okay, double stranded of nucleotide. So this is called what the double helix or double stranded DNA. So this is the one that carry the gene or genetic material. So bacteria has this chromosome with DNA. Okay, and then there is another genetic materials that present in bacteria, and this is only present in bacteria, which is called the plasmid. This plasmid actually smaller than the chromosome, is also circular with DNA over here. Okay, so plasmid also carry genetic material, mostly for resistant gene. Okay, so carry the resistant gene. Ribosome, again, all cell will have ribosome, including this bacteria or prokaryotic cell. The difference between ribosome in prokaryotic and eukaryotic cell are the size. So ribosome of prokaryotic cell is smaller, which is 70S. That's the size unit. While for the eukaryotic cell, it has 90S. So eukaryotic cell has bigger ribosomes. The main function of ribosome is for making protein. So this is the site for protein synthesis. Now the special structure inside the bacterial cell is called the inclusion and granules. So this mostly is used for storage, okay? Storage of molecule, like extra molecules, maybe carbohydrate, maybe lipids will be stored in this inclusion and granules. So whenever the condition is not favorable, for example, there is not enough nutrient, then they can use the storage of food in this inclusion and granules. The cytoskeleton, this is skeleton of the cell. Okay, so skeleton, cyto means cell. Okay, skeleton is the skeleton. So this is the skeleton of the cell, which is consists of network of thin proteins. Sometimes it's called the filament. Okay, that present inside the cell. And the last one, which is the special structure of only some bacteria that have this endospore. which is a group of Clostridium bacteria, Bacillus bacteria. This is the two main bacteria that have endospore, and also there is another group of bacteria called the Sporosarcina that also have the endospore. So bacteria that produce endospore, they will have two life cycle, which is they will have vegetative cell. Okay, so during the normal condition, okay, so normal condition, they will have the vegetative cycle. So this is just a regular type of cell where they reproduce. Yeah? But in a very, very difficult situation, like for example, very dry or very hot, right? These bacteria are able to produce what we call the endospores. This endospore will protect them against any adverse environmental condition. Even they are able to stay in that condition for millions of years. Okay, so endospore actually can protect them for a long, long time period, okay, during the difficult conditions of their environment. So it's very long survival protection with the endospore. Again, this is the cycle. During normal condition, they will in the form of vegetative cell, right? Which is this is again, this is the normal cell in normal condition. So during this time, they're able to reproduce and etc. But if the condition is not good, okay, if a very, very difficult, maybe very dry or very cold, for example, then they start to making this cycle, which is producing the endospore. So this is the endospore over here. If you see, the cell is actually really, really, is the cell over here protected by a very thick layer, eh, that protect the cell from any bad condition of weather or environment. So when the condition is favorable again, then they can hatch into vegetative cell. Okay. So again, in this condition, they can stay for a very long period. As I mentioned before, it could be million year. Okay, even they can stay in this condition for million year. Okay, so this is a special structure of some bacteria, which is endospore. So they have two type of life. Okay, so which is the vegetative type and the endospore type. This endospore actually will cause them to be very difficult to destroy, to be destroyed. So when we try to kill this bacteria like Clostridium or Bacillus bacteria, then we have to perform a process is called the sterilization. Okay, so this sterilization will be able to destroy this endospore. Regular cleaning methods like disinfectant, for example, or boiling water will not kill this endospore, will not destroy the endospore. So it needs special methods. It's called the sterilization. Example of sterilization is using autoclave. Okay, in many lab or clinic or hospital, they will have autoclave in order to sterilize their equipment. So this is actually the process that will also kill the, of course, kill the vegetative cell and also destroy the endospores. I think that enough for this lecture. Next, we will continue with the bacterial cell shape and arrangement, including also some classification of bacteria. Goodbye now. Hi everyone, welcome to medical microbiology. So today we will continue chapter 4. This is going to be the last lecture of chapter 4, structure of bacterial or prokaryotic cell. So today we're going to discuss about the shell shapes, arrangement, the size, and also classification of bacteria. Shell shape, arrangement, and size. Remember when we talk about shape, shell shape over here is actually cell. Okay, so this is for cell, which is we have to see them under the microscope. Don't be confused with the colony shape, because colony shape we able to see them with our naked eyes, which is we see the colony shape on the culture media, on the petri dish. Okay, when you have a bacteria growing on the petri dish, then you will be able to see the colony, and that colony also have shape and size, color, and etc. But today we're going to learn about cell shapes, which is we have to see it under the microscope, because we cannot see the bacterial cell without the microscope. So by using the microscope, then we should be able to see the three main type of shell shapes. The first one is called coccus. Okay, coccus bacteria is mean round or spherical bacteria. Okay, like this one over here. This is a coccus bacteria. And if you see, sometimes they also join together. So many cell, okay, they bind together to make this group of cell. Okay? But each individual cell over here has round shape. So this is what we call the coccus bacteria. The second one is called bacillus. Bacillus means rod. Okay, it look like a rod, like this one. Is a bacillus bacteria over here is rod. Okay, this is under microscope how it look like. Okay, this is the bacillus bacteria. Sometimes they're also grouping together, sometimes one by one. But again, each individual cell will have rod shape, and this is what we call the bacillus bacteria. The next one, the third one, is called the spirillum, which is kind of twisted or a spiral bacteria. Okay, like this over here. You see that this is like a spiral. This is twisted spiral also. So this group over here is called the spirillum bacteria. Okay, spirillum bacteria. But there are also some bacteria that their shape is between one of these three main shape. Okay, like for example, between spirum, okay, spirillum and rod, there will be like coma bacteria over here. Okay, it's called the fimbrial. So between spiral and bacillus. Sometimes between round and bacillus, there is kind of oval bacteria. Okay, so again, there are three main shapes of bacterial cell: coccus, bacillus, and spirillum. Between them, there are other kind of shape like coma shape, yeah, oval shape. Okay. The arrangement, as you see before under the microscope, sometimes coccus bacteria, they are just single. So just one. Individual bacteria separated one. Or sometimes they combine, become two. It's called a diplo. D mean two, so it's going to be Diplococcus bacteria. Or maybe making like a chain bacteria over here. Okay, that chain bacteria, this one over here, is called strepto. Okay, so if you see streptobacillus or streptococcus bacteria, the one that causing the strep throat. Okay, so this is it has the arrangement like chain. So streptobacillus. Sometimes they make four. Okay, it's called a tetrad, like this. Or sometimes make cubicle, is called a sarcina. Okay, this is sarcina bacteria. Sometimes it's make irregular cluster, like this. So the irregular cluster is called staphylo. Okay, so if you heard about staphylococcus bacteria, staphylococcus aureus, for example, that bacteria has round shape with arrangement of cluster, like bacteria that live in our skin, Streptococcus aureus. It has a cluster arrangement. And then the same thing with the bacillus bacteria, maybe Diplobacillus, which is two rod bacteria, or maybe Streptobacillus. It has long chain of bacillus bacteria, or palisade. This again, this is going to be called Streptobacillus. Palisade is look like fence. Like they arrange like this. So this is individual bacillus bacteria that combine together to make like a fence. Okay. Now the size. Bacteria is really small. We cannot see them without microscope. Now we compare the size. Let's say this is our hair. So you can see your own hair, right? It's very, with your naked eyes. But this is over here. This is other group of organism. Like pollen, maybe you can see it. Lymphocyte. And this is white blood cell. Really small. Actually, you have to see it under the microscope. This is yeast, which is fungi, which is smaller than our white blood cell, the lymphocyte. This is red blood cell over here. It's also a little bit bigger than the yeast. And then this is bacteria over here. Okay, so that's the reason one bacteria sometimes able to enter our cell, because they are really, really small. So when we make it bigger, then this is one of the biggest bacteria, the E. coli. Okay, but even though this is the biggest cell, but still we unable to see this bacteria with our naked eyes. Okay. This is Staphylococcus bacteria. Okay. With each of these individual is in round, is coccus bacteria. This is virus. Virus is really, really small. Most of the time we will not be able to see them using the light microscope. We need the electron microscope to see these viruses. Okay, so this is the kind of comparison between size of several microorganisms. Yeast or fungi is bigger then bacteria is smaller, and viruses are really, really small. No classification. Okay, for the classification, there are several characteristics that we use to classify bacteria. First, microscopic morphology, which is using the microscope. So this one, we can say what is the cell shape? Maybe coccus, bacillus, okay, or maybe spirillum bacteria. Okay? Microscopic. This is the one that we see with our naked eyes. Colony, maybe colony like round or circular. Okay, maybe the color yellow, tan color, for example. Okay? So this is the one that we see with our own eyes, our naked eyes, the colony of bacteria. So this colony means it has million of bacteria that make this colony. So colony consists of million of bacteria. Therefore, we can see using our naked eyes. The bacterial physiology, okay, and also using the serological analysis, using the antigen antibodies complex, okay, reaction, and check their DNA, the genetic, yeah, maybe using the PCR, for example. Then from that, okay, the classification that used for bacteria, the most famous one is using this, what we call the Burgess manual of bacterial taxonomy or classification. Again, when we talk about the classification for bacteria, it included in a group of archaea or bacteria. Remember, archaea is bacteria that live in extreme environment. Maybe in hot spring water, very salty water, for example, or very salty environment. This is archaea. So most of the archaea will be free living. Okay. And the second domain will be bacteria. This is regular bacteria. Some of them may be free living, maybe parasite, okay, or causing disease to us. Sometimes also known as pathogenic bacteria. Okay, so some of them may be causing disease to human, other animals, plants, etc. So this is the second domain. Okay. Remember, there are three domains: archaea, bacteria, and eukarya. So other organisms will be included in the eukarya, okay, including animals, plants, algae, fungi, protozoa will be in eukarya domain. Okay, so this is the kind of summary of classification of organism. So this is the whole organism in this world will be classified into this group. Again, the first domain is archaea. Okay, archaea will be consist of these free living bacteria that live in the extreme, okay, extreme environment. Okay, and then the second group will be bacteria that consists of this phylum, okay, so many of them, Some of them free living, some of them causing disease or pathogenic. And then, okay, this is the first domain, this is the second domain, and this is the third domain, which is eukarya. And eukarya will consist of protozoa and algae. Okay, this one is called, both of them is called protists. Okay, so this is the protists, and then plant, okay, animals and fungi. Okay, slime mold is also between kind of fungi, animals, and plants. So they all have real nucleus. So this one have nucleus. These two no nucleus. Okay, so this is just a summary of this classification. We will learn this more on the next chapters. Okay? Like for example, for fungi will be in chapter 22, then worms like animals, it will be in chapter 23. Okay, protozoa and algae, I believe this is also in chapter 23. Okay, protozoa, algae, animals or worms in chapter 23. And bacteria classification will be mostly in chapter 18, 19, 20, and 21. So we will learn this later. Okay, so right now it's just kind of introduction of this classification. And I think it is done. So chapter 4, we finished chapter 4. We will continue with chapter 5 on the next lecture. Goodbye. Hi everyone, welcome to medical microbiology. So today we will study chapter 5, survey of eukaryotic microorganism. For the first lecture, we will learn about the external structure and the cell boundary of eukaryotic cells. According to some scientists, okay, the eukaryotic cell were evolved from prokaryotic cell. Just remember that eukaryotic cells are bigger than prokaryotic cells. And remember again, prokaryotic cells are bacteria. Other organisms will be eukaryotic organisms like protozoa, algae, plants, fungi, animals. They all eukaryotic organisms. So according to some scientists, this eukaryotic cell, they are developed from the prokaryotic cell. According to them, these prokaryotic cells, they perform what we call the symbiotic association. So let's say this is bigger prokaryotic cell, so one bigger prokaryotic cell, and there is a smaller prokaryotic cells over here. So these two cells, they perform symbiotic association, where the smaller one enter into the bigger cell and stay inside the cell, and finally it's become organelle. Okay, it's become organelle. So this is become eukaryotic cell. Okay, so this is the development or the evolution of eukaryotic cell that actually evolved from the prokaryotic cells. Now, eukaryotic organism, because we're talking about microbiology, so it's gonna be the microorganism over here. So we will not learn plants or bigger animals. We only learn the microorganism, the smaller eukaryotic organism. Like for samples, there are some of them that are unicellular, okay, unicellular organism like protozoa. What is unicellular mean? Unicellular mean one cell is become one organism, and this cell able to perform all characteristic of life like reproduction, metabolism, respond to the stimulus, and etc. Okay, so this is what we call the unicellular organism. Some of them may be colonial, maybe multicellular, and some still unicellular, like fungi and algae. Now, what is the difference between unicellular, colonial, and multicellular? Again, for unicellular, one cell is become one organism. They perform all the characteristic of life. This is unicellular organism. For the colonial organism, is organism that is made of several cells that combine together. Okay, make a colony. Okay, when we separate one cell from the colony, this cell still alive. Okay, this becomes still live cell, and this cell will be able to combine with other cell to make another colony. Okay, so this is what we call the colonial organism. They're able to combine with other cell, but they're also able to live by themselves. Okay, this is the colonial organism. While for the multicellular organism, this is the organism that consists of many cells, million of cells, like human, for example, or animal or plant. They consist of million of cells. Okay? If this one cell is taken from the multicellular organism, this cell will be die. Okay, it will not be alive like this cell in the colony. Right? So this is multicellular organism. Okay, so that's the difference between unicellular, colonial, and multicellular organism. So for the multicellular organism that we will learn in this class, mostly helminths, which is the parasitic worm. And some fungi, they are also multicellular. We will also learn a group of animal that is called as arthropods. This is a group animal that carry disease, like for example, mosquitoes, okay, tick, fleas, lice. This is a group of animal that is called the arthropods that able to transfer disease from uh, one organism to another organism, from one patient to another patient, for example. Now, if we look at the structure of eukaryotic cell, it is actually similar with the prokaryotic cell. It is similar with the bacterial cell. It has three main structure, which is the external structure, the cell boundary or cell envelope, and the internal structure. For the external structure, some eukaryotic organisms have appendages, which is used for movement like flagella and cilia. Okay, And also some eukaryotic cell, they also have glycocalyx that could be capsule or slime layer. And the cell boundary, eh, which is some of them have cell wall and cell membrane. Okay, like for example, plant, uh, in this case, because this is microorganisms, gonna be algae, okay, and fungi. So algae, fungi, plant, they do have cell wall and the cell membrane. Uh, for protozoa and animal, like worm, for example, they only have the cell membrane, okay, without the cell wall. Now for the internal structure, there are some structure that's similar to prokaryotic cell, which is the cytoplasm, uh, ribosomes, and the cytoskeleton over here. Okay, these are the internal structure that's similar with the prokaryotic cell. The nucleus, ER, whether RER or SER, Golgi apparatus, lysosome, vacuoles, mitochondria, and chloroplast. This is only present in eukaryotic cell. The prokaryotic cell do not have this organelle. Okay, this is the organelle that only present in the eukaryotic cell. Now we start with the external structure. Okay, especially there is an external structure that is called the locomotor appendages, which is the appendages that used for movement. This is usually present in a group of eukaryotic organism is called the protozoa. So protozoa has flagella, which is the structure that used for swimming. Okay? The way this protozoa move is by whipping the flagella. If you still remember, the bacteria also have flagella, but the way they move is by rotate. Okay, by rotating the flagella. Okay, so that's the difference between the eukaryotic cell and prokaryotic cell. This is bacteria. Okay, so by rotating the flagella, if it is counterclockwise, then the bacteria will move forward. If it is clockwise, then the bacteria will be tumble. Okay, remember that. And then for the eukaryotic cell, the way they move is by whipping this flagella. Another locomotor appendage is called the cilia, and this is also mostly present in protozoa. Okay, so protozoa has cilia, is hair-like structure over here. Okay, that is used for movement. The way it's moved by like a swimming, or a power stroke. So they live in water and then stroke their cilia to move. Okay, the next external structure is glycocalyx, and again this is only present in some of eukaryotic cell, like for example some fungi have this glycocalyx, whether capsule or the slime layer. Under the glycocalyx there will be cell wall. Okay, so cell wall. Okay, so fungi and algae, they do have cell wall. Okay? While protozoa and animal like helminths, they do not have cell wall. Okay, lack of cell wall. Now, when we're talking about the cell wall, there are a little bit different between fungi and algae cell wall. Okay, just recall the bacteria cell wall. Okay, so bacteria cell wall on the previous chapter, right? The bacteria cell wall consists of uh, peptidoglycan. Okay, remember that. If the peptidoglycan thicker, it's become gram-positive bacteria. If it is thinner, it's gram-negative bacteria. Okay. Now, for the eukaryotic cells, okay, for fungi, the main structure of their cell wall is called chitin, which is mostly protein. Okay? While the algae and plant, their primary molecule on their cell wall is called cellulose, which is carbohydrate. Okay, now look at the peptidoglycan over here in the bacterial cell wall. Consists of protein and carbohydrate. It's a combination of protein and carbohydrate. Fungi cell wall mostly consists of protein. The algae cell wall mostly consists of carbohydrates. Then under the cell wall, okay, there is a cell membrane. Cell membrane. And the cell membrane of eukaryotic cells and prokaryotic cell will be the same, which is, remember, phospholipid bilayer. Two layers of phospholipid molecules, and of course there are also some proteins on it and cholesterol or sterols on the cell membrane. I think we done with the external structure and the cell boundary. We will continue with the next lecture of the internal structure of eukaryotic cell. Goodbye now. Hi everyone, welcome to medical microbiology. Today we will learn about survey of eukaryotic microorganism, which is chapter 5, and today we are in lecture number 2 of chapter 5, which is we will discuss about internal structure of eukaryotic cell. Again, we will review the structure of eukaryotic cell. It has three main structure, which is the external structure. Okay, the external structure may be appendages like flagella and cilia over here. So this is flagella, glycocalyx, maybe capsule, and slime layer. Just for information, not all eukaryotic cells have this flagella, cilia, capsule, and slime layer. Some of eukaryotic cells do not have this external structure. The second part is the cell boundary, which is consists of cell wall and the cell membrane. Now just remember again, the one that have both cell wall and cell membranes are algae, fungi, and plant. Okay, but we do not discuss plant in our medical microbiology because plant are bigger organism. Then the one that only have cell membranes are protozoa and animals. Okay, so cell membrane, protozoa and animals only have cell membranes, no cell wall. And then the last part of the structure will be internal structure, which is the structure inside the cell over here. Okay? Which is the first one will be the content of the cell, which is the cytoplasm. The next one will be nucleus. This big one over here is a nucleus. And then ribosome. Ribosome is actually small organelle, like a dot over here. So that's the ribosome. And then ER, there are two types, RER and SER. The RER is the one that have ribosome on it. So therefore, they are rough ER. And then the SER is this one over here, the one that do not have, okay, this is SER, the one that do not have the ribosome on it. Therefore, they are smooth. Okay? And then Golgi apparatus, which is this one. Okay? And then lysosome. Lysosome is like organelles, smaller organelle that carry enzyme is called the lysozyme. And then some vacuoles, mitochondria. Mitochondria is this one is really unique. It has two layers of membrane, inner and outer layers. And then chloroplast. Chloroplast is also here, very unique. It has green color. And then cytoskeleton, which is the filament inside the cell. Now we will discuss one by one this internal structure. We're gonna look at the function also first. Okay, nucleus. This is the one that carry the genetic materials. So this is the genetic center of the cell. It's carry the DNA. And also this nucleus will synthesize RNA. So RNA are synthesized inside the nucleus. In the nucleus, there is also smaller structure is called the nucleolus. This is the place for making ribosome. Okay, ribosomal RNA synthesis. So the ribosome will be made by this nucleolus. Okay, so this ribosome over here is actually made by the nucleolus. And then ER, okay, endoplasmic reticulum. There are two types, RER and SER. Okay, the function is to transport material and also for lipid synthesis. This is for SER. So SER makes lipids. And then Golgi apparatus. This is the next bigger organelle used for packaging and modification of protein and also lipids. Okay, for productions of new product. And this new product sometimes stay inside the cell, sometimes it is secreted outside the cell. And then the next organelle is called the lysosome. Lysosome is actually made by the Golgi apparatus. It has enzyme that used for digestion. And then vacuoles, it is a place for storage, mostly in plant, okay, plant algae. They have bigger vacuoles. Animal cell, usually they do not have vacuoles. If they do, they usually have smaller vacuoles. And then mitochondria, this is the place for making ATP. Okay, so energy production, which is actually ATP, and ATP made from glucose and oxygen. There are three processes of this ATP synthesis, which is using the uh, Krebs cycle, electron transport, oxidative phosphorylation, and also actually there is a process called the glycolysis. Okay? And then chloroplast, this is only present in algae and plant. Okay, this is the organelle that used for making chemical energy, which is mostly glucose using the light. Okay, therefore the process is called the photosynthesis. So this is synthesis of glucose using light. Photo mean light. And then ribosome, this is the place for protein synthesis. And then cytoskeleton, this is the filament, eh, which is microfilament and also microtubules that provide the cell structure. So it's a kind of keep the shape of the cell, and also this filament and microtubule is used for movement of some molecules inside the cell. Now we start with the first internal structure, which is nucleus. This is the center of the cell, and this nucleus carry the genetic materials, which is located in the chromosome. Okay, so in chromosome there is a molecule called DNA, which is the genetic materials. So during the resting stage, usually the chromosome will form like a thread. Okay, look like a thread. This is what we call the chromatin. So this is chromatin. Okay, so chromatin is actually the chromosome that is not thick yet. Okay, when the cell is ready for cell division, then chromatin will change into chromosomes. And there is also here, this is nucleolus. This is the place for making the ribosome. Okay. Okay, this is the synthesis of ribosomal RNA, which is nucleolus. And if you see over here, the envelope, the nuclear envelope, it has pore. Okay, why? Because remember the DNA over here that is carried by this chromosome will be transcribed into RNA. Okay, and this RNA then have to go outside from the cell. Right? They have to go out from the cell in order to be translated into proteins by the ribosome. So the pore is used for transportations or exit of the RNA from nucleus into the cytoplasm. Now the second structure is the ribosome. Okay, so ribosome, it has two units, the smaller unit and the bigger unit. Okay, and this is ribosome of eukaryotic cell. So the ribosome of eukaryotic cell is bigger, which is 90S. Okay, 90S. Remember for the prokaryotic is 70S. Okay, so prokaryotic cell has 70S ribosome, which is smaller. Now, what is the functions of ribosome? The ribosome used for making protein. So protein actually made from messenger RNA that is made inside the nucleus. This messenger RNA will exit the nucleus, go to the cytoplasm, and then in the cytoplasm, it will be trapped by this ribosome, and then this messenger RNA will be translated into this protein. Okay, so this is protein. So protein is actually made from messenger RNA by the ribosome. Now, if you see this organelle, this is the RER. Okay, this is the rough ER, rough endoplasmic reticulum. Why it is rough? Because there are so many ribosomes attached on its wall. So whenever the protein is made by the ribosome, then it will be stored. Okay, it's storage over here. Yeah, it will be stored inside the lumen of this RER. Okay, so this is the lumen of RER, which is used for protein storage. Okay, so now you know the reason why the RER have a lot of ribosomes on it, because ribosome is used for making protein, and then that protein will be stored in the RER lumen. Okay? And then if you see the location of this ribosome and RER is very close to the nucleus. So this is nucleus over here. See that one? The nucleus make the RNA, which is messenger RNA. Okay, it is made from, let's say, I'm using this for DNA, right? So DNA will be made into RNA. Then RNA will go out from the nucleus using this spore. Okay, and then will be available here. Will be available for the ribosome to make, in this blue color, okay, to make the protein. So protein will be located in this lumen. So again, if you see this location, okay, and the structure, we know that the structure telling us that structure fits the function. Why, for example, RER is very close to the nucleus. Why there are ribosomes on it? Because as soon as the messenger RNA leave the nucleus, it will be attached by the ribosome, and then it will be converted into the protein, and the protein will be stored in this lumen of the RER. Okay. So after the protein is made and stored in the RER lumen, then it will be carried by this transport vesicle into this Golgi. So this is actually Golgi apparatus. Okay, inside the Golgi apparatus, then the proteins will be converted into the final product, like enzymes or hormones. Okay, so the Golgi apparatus will be the place to make the final product. Now we look at the next structure, which is Golgi apparatus. This one over here. Again, this Golgi apparatus will be used for packaging the protein and to make the final product like enzyme and hormones. Okay. If you see another membrane. Okay, another organelle over here is called the SER. Okay, smooth ER. This smooth ER is used for making lipids, and this lipid will also carried into Golgi apparatus. So basically, protein will be carried into Golgi apparatus. The lipid also will be carried into the Golgi apparatus by this vesicle transport. Okay? And inside the Golgi apparatus, then it will be made into final product like enzyme or hormone. Okay. So again, this is the Golgi apparatus, which is the organelle that used for modifying the proteins and lipids into enzyme, okay, or hormones. Okay, look at what happened here. Again, we start from nucleus, yeah, the nucleus over here and nucleolus over here. Okay, so nucleolus produce the ribosome. Okay, the blue one over here is the ribosome. It's made in the nucleolus, and in the nucleus, the DNA, okay, will be converted or transcribed into RNA. Okay, so this is DNA. Then they also will go out from the cell, from the nucleus into the cytoplasm, where they will be translated into protein. Okay, it will be translated into protein, and then the protein will be stay in this RER lumen. And then this protein, so let's say over here, it will be carried by the transport vesicle into this Golgi apparatus. And then in the Golgi apparatus, it will be made into different products. Okay, maybe enzyme. Okay, so the different product over here, maybe enzyme or hormone. Okay, if it is hormone, they usually will be secreted from the cell because the hormones will be delivered to other cell in the body. Like for example, if it is human cell, that hormones over here will be produced by one type of cell is called the gland. Let's say for example, estrogen. They are produced by the ovary, which is located in the pelvic area of the aminopelvic cavity. And this estrogen will be excreted from the ovary into different location. Okay, maybe go to the, for example, to the mammary gland, okay, to the hip area, and etc. Okay, that will create the female secondary characters. Okay, so again, if it is hormone, mostly it will be transported out from the cell. Some enzyme, they will stay inside the cell. So the one that transport the product from Golgi apparatus into outside is called the secretory vesicle. Okay, so there are two types vesicle. The first one is called the transport vesicle from the RER into Golgi apparatus or from the SER into Golgi apparatus. And then the secretory vesicle and condensing vesicle is the one that carry the product from Golgi apparatus to outside the cell. Okay, so transport vesicle and condensing vesicle. Okay, the next organelle is lysosome. Okay, lysosome is actually made from Golgi apparatus. So, like for example, enzyme that is made by the Golgi apparatus, some of them stay inside the cell and usually carried by this lysosome. So that enzyme is called the lysozyme. Okay, so lysozyme, let's say this is lysosome, then lysozyme will be here. Okay, this is lysozyme and this is lysosome. Okay, so the organelle that carry lysozyme is called the lysosome. Now, what's the function of lysosome? It is used for destroying foreign materials that enter into the cell. It could be maybe food or maybe bacterial cell or maybe viruses. Okay, so foreigner cell that enter into the cell, right? Foreign molecule that enter into the cell will be destroyed by this lysozyme that is carried by the lysosome. Now, when the lysosome combine with the foreigner particle that enter into the cell, then the organelle become what we call the phagosomes. Okay, phagosome is the combination between lysosome and the foreigner particle that enter into the cell. Another organelle internal structure is vacuoles, and again mostly this is in plant and algae. Okay, the next one is mitochondria. Mitochondria is the organelle that used for making energy, which is ATP. Okay, so ATP is made by this organelle, and this organelle really uh, specific because it has double layer. The outer layer is smooth, okay, and the internal layer is actually making curve. It's called the cristae. Okay, up and down, windy, winding kind of layer over here. Right? So that's the internal layer. It's called the cristae membrane. And again, this is the place for making the ATP. Now, if you look at this mitochondria, it also carry the chromosome. Remember when we talk about the evolution of eukaryotic cell? Eukaryotic cell is considered evolved from two prokaryotic cell that making the symbiotic association. The bigger cell actually kind of take smaller prokaryotic cell. So this mitochondria is considered as the smaller prokaryotic cell that stay inside the bigger prokaryotic cell. Therefore, it has chromosome over here. It's carry the chromosome. This is as a proof that eukaryotic cell are combination of two or many prokaryotic cell. This one is chloroplast. So chloroplast, again, chloroplast only present on the eukaryotic cell that able to perform photosynthesis. Okay, this is used for photosynthesis. Now, which cell able to perform photosynthesis? Of course, the cell that carry the green pigment, which is plant. So plant are green, right? And also algae. Okay, there are many green algae that are able to perform photosynthesis. So this chloroplast only present in this eukaryotic organism, plant and algae. If you see the chloroplast consists of stack of thylakoid membrane. So the one membrane over here is what I call the thylakoid. This is another thylakoid, and the whole thing is called the grana. So the whole thing, okay, one stack of thylakoid membrane is called the grana, and this is the one that carry the pigment is called the chlorophyll, which is green pigment. Okay, why is green pigment? Because they actually do not absorb the green color. They reflect the green color. That's the reason why we see green, because of reflection of green color by this chlorophyll. So what is that mean? It means that the green color is actually not used for photosynthesis because it is reflected. Which spectrum of light that used for photosynthesis? Mostly the blue color and the red color. Okay, the blue color and the red color. The green color is reflected. Okay, and also in the chloroplast, you will find the yeah, in the chloroplast you will find this chromosome or DNA. And again, this is used as a proof that this chloroplast maybe during the evolution is actually smaller prokaryotic cell that stay inside the bigger prokaryotic cells. The last one will be cytoskeleton. And the cytoskeleton is the same like skeleton, which is rigid molecules that give the shape of the cell. Just like, skeleton, the bone of our body is actually give the shape of our body. Okay, so the same thing with this one. This skeleton also give the shape of the cell. It consists of two main proteins. The first one is called the microfilament, and the second one is called the microtubules. Tubules is bigger, so this is microtubule over here, which is bigger molecules of protein. Microfilament is smaller, and also it actually divided into two types. The very thin one is called actin. Okay, so actin means very thin, and then the middle size is called the intermediate filament. Okay, basically there are three types of cytoskeleton: actin, intermediate filament, and the microtubules. Okay, I think we're done for lecture 2, chapter 5. We will continue lecture 3, chapter 5, which is we're going to start with the first type of eukaryotic organism, which is fungi. I will see you then, and goodbye. Hi everyone, welcome to medical microbiology. So today we will continue chapter 5. Today lecture 3, we will discuss about fungi. Okay, we will discuss about fungi, one of the eukaryotic organism. Okay, so remember, eukaryotic organisms are organisms that has nucleus and internal organelles. Okay, there are several of them: fungi, algae, protozoa, and parasitic worms, okay, or helminth, that will be learned in this class. Okay, just for your review, remember again, all organisms in this world divided into taxon. Okay, it's taxonomy or classification. Remember, the highest taxons will be domain and kingdom. And there are 3 domains. Remember, 3 domain of organism. The first one is called domain archaea. The second one is called domain bacteria. Okay, and the last one is called domain eukarya. Okay, and there are 5 kingdoms, right? 5 kingdom of organism. Okay, the archaea and bacteria, they are prokaryotic cell. They do not have nucleus, do not have organelles. Okay, they are in one kingdom. The kingdom is called kingdom Monera. Okay, this is for all prokaryotic cell. The other 4 kingdom will be the one that have nucleus, which is under the domain of eukarya. So will be the kingdom of fungi, then kingdom of protists. Okay, and protists divided into 2 types, algae and protozoa. Okay, and then the next kingdom will be kingdom of plant. Okay, plantae, and the last kingdom will be kingdom of animalia. Okay, so all of these kingdom over here, 2, 3, 4, and 5, they are eukaryotic cell. Therefore, today we will start with the first kingdom, which is the kingdom of fungi. And on the next lecture, we will talk about algae and protozoa, which is under the kingdom of protista. And then helminths or parasitic worm, which is under the kingdom of animalia. We will not talk about plant or plantae because they are mostly macroorganism, big organism. Okay, remember our class is medical microbiology, which is smaller organism. Okay, we start with the kingdom fungi. Kingdom fungi itself, there are 2 main types, which is the first one is the microscopic fungi. It means that we can see them. For example, mushroom, puffballs, gill fungi. So we able to see them with our eyes. So they are microscopic. We will not learn this fungi in our class because they are microscopic. The one that we will study in this class is the microscopic fungi. This microscopic fungi, there are 2 types, which is hyphae. So it has long filament on their structure. And sometimes if they combine together, we can see them. Their colony is called molds. So molds is actually hyphae type of microscopic fungi. The second one is yeast. Okay, this is more individual short fungi. So these are the 2 type of microscopic fungi that we will learn in this class. Okay, this is the illustration of 2 different type of microscopic fungi. This one over here is the hyphae. Okay, hyphae, the one that has long filament on their body. And then this one is the yeast. Okay, it's short body. Sometimes it's only one individual cell, sometimes two or three, but it's not long like this hyphae. Okay, if you see the hyphae, then there are two types of hyphae. The first one, the one that each cell has border, has septum. Okay, so therefore this type of hyphae is called the septate hyphae. Okay? If you see under microscope, it's gonna be like this. This is the septum, the border that separate two cells, and each cell will have one nucleus. The other type of hyphae is non-septate. Okay, there are no septum between the cells, so this is just one long cell. This is one long cell. Because they are long cell, then they have many nucleus in one cell. So a lot of nucleus, okay, in one cell. The yeast, yeah, they're gonna be short cell, right? So it's only one cell will have one nucleus. Okay, so this is the hyphae. Okay, so if you see under the microscope, it's gonna be look like this. This is under microscope. Okay, you see this hyphae. Okay, so long hyphae, and this also have septum. So this is septate hyphae, right? But if you see with your naked eyes, it's gonna be like this. This is actually colony of this fungi, and this is what we call molds. Okay, sometimes you see molds on the top of the bread, or if you drink coffee, if you don't need, if you leave it for several days, then molds will be growing on that food or coffee. So this is mold, which is if you take sample and then see it under the microscope, it's gonna be like this. Okay, this is the yeast. Again, mostly individual cells. Sometimes they have 2, 3, 4, or 5 cells, but it's very short. Okay, very short filament. And that's what we call the pseudo hyphae. Pseudo means fake. Yeah, this is not real hyphae. Okay, and this is if you see under microscope. It's gonna be look like this. Look like a grape. Okay, the fungal nutrition, they are heterotrophic. Hetero mean others. Okay, trophic mean food. Okay, so it means that these fungi have to get food or nutrition from other. So they have to take it from other. They're unable to make their own food. So all organism that unable to make their own food is called a heterotrophic. Okay, heterotrophic organism. Okay, this including animal like human, because animal cannot make their own food. They have to eat, right? Eat other organism. And then fungi, okay, fungi, they have to absorb. Okay, they have to absorb the nutrient from that organism. Protozoa, protozoa, this is also heterotrophic, so they also have to eat. And then, of course, bacteria. Bacteria also heterotrophic organism, so they have to absorb. Okay, sometimes many of them become parasite. Okay, they have to take the nutrients from their hosts. The opposite of heterotrophic is called autotrophic. autotrophic. Okay, auto means self, trophic means food. So this organism able to make their own food. Example are algae and plant. Okay, this organism able to make their own food using process called the photosynthesis. Okay, so sometimes autotrophic organism is also known as photosynthetic organism. They're able to make their own food. Okay, so again, fungi will be heterotrophic, so they absorb the nutrient from other. That's the reason why many fungi will be also parasite, which is take the nutrient from their host. And the disease that caused by fungi is called mycosis. Okay, this is example of mycosis over here, which is the skin infection caused by fungi. If we take sample over here and then see under the microscope, it's gonna be look like this. Okay, so you see the septate hyphae okay, under the microscope, and this is spore. Actually, you see spore. Okay, so these examples of mycosis, which is skin infection that caused by fungi. And again, many heterotrophic organisms, if they are small, many of them become parasite, like bacteria, these fungi, some protozoa is become parasite. Animal like, worms, the parasitic worm, they are become parasite. Because what? Because they have to take the nutrient from other. And again, this is what we call the heterotrophic organism. Right? This is how the fungi reproduce. They produce structure is called the spores. Okay? Spore is very similar with maybe seeds, okay, in plant. Okay, so they are produced by a structure and then released into the environment, sometimes carried by the wind, okay, and then drop on other place and it is growing into new individual, and that is going to be spread like this. So this is how this fungi reproduce, which is using the spores. There are two types of spores. The first one is called the asexual spores. Okay, A mean non. Okay, so this is gonna be non-sexual spores. The spore that is not produced by mating process, not produced by sexual process. Okay, there are two types of non-sexual spores. The first one is called the sporangiospores. Is a very straightforward. The sporangiospore produced by fungi that has sporangium. What is sporangium? Sporangium is a sac-like structure. Okay, it looks like a pocket over here, with cover. Okay, and then inside the sporangium there are a lot of spores in it. Okay, so this spore that produced from the sporangium, then it is called the sporangiospores. Okay, very easy. The second one is called conidia. Okay, so, or conidiospore. The shortest name is conidia. Conidia is the non-sexual spore produced by other structure than sporangium. Maybe structure like this, and this is become conidia. So maybe it has several names, but their general name is called conidia, which is the non-sexual spore that is produced by other than a structure like sporangium. Okay, so this the same thing. Yeah, there is no coverage over here. There is no sac. Okay, therefore it is conidia. This one is conidia, conidia. This is conidia. all of these are conidia. So this is the second type of non-sexual spore, sporangiospore and conidia. Now the second type of spore is called sexual spore. Sexual spore is the spore that produced by process of mating. So there is mating process. Okay, for fungi, we cannot really say male and female or boy and girl. Usually just call positive and negative hyphae. Okay, so these positive and negative hyphae, they can perform mating over here. You see that? They combine, and finally what happened? They growing, and the spore that produced by this site is become sexual spores. Okay, that's the sexual spore. There are three types of sexual spore. First is called the zygospore. Okay, it is produced by zygote, which is fertilization of positive and negative hyphae, and then produce this spore like a zygote. This zygote is actually spore. So this is what we call the zygospores. Usually this is for the mold that grow in bread, for example. They produce the zygospores. Okay. And then the second type is called the ascospore. Okay, it is produced by this mating process, and from this mating process, they make a structure is called the ascus. Okay, ascus. Therefore, the spore that produced by this structure is called the ascospore. Okay, ascospore. That's the second type. The third one is called the basidiospore. Okay, this is on gill fungi, for example. Usually the positive and negative hyphae, they mating in the under the ground, okay, in the soil, they're mating together over here, and then finally growing into gill fungi over here. And then what happened under this, there will be structure. It's called the basidium. Okay, if it is many, it's called basidia. And then this basidia or basidium will produce the spore, and the spore will be called as basidiospores. Okay, so these are the three types of sexual spores of fungi. Now, based on this sexual spore, okay, then fungi can be classified into several phylum. Okay, remember after kingdom, okay, there will be phylum. Okay. So there are 5 phylum of kingdom fungi. The first one is called the phylum Zygomycota. From the name, then you should be able to guess this phylum will produce zygospores. Okay? Zygospore. They also produce the sporangiospores, and some produce conidia. So this zygospore is sexual spore. Okay, this is sexual spore. This is non-sexual spore. Okay, so this zygomycota produce sexual spores is called zygospore, and non-sexual spore is called the sporangiospores. Okay, and then the second phylum will be Ascomycota. Again, from the name, then you should be able to guess this phylum consists of fungi that produce ascospore as their sexual spore. And they also produce the non-sexual spore is called conidia. So produce both, okay? Produce sexual spore and non-sexual spore. The next one is going to be Basidiomycota. And from the name, again, you should be able to guess that this phylum consists of fungi that produce basidiospore as sexual spore. They also produce the conidia as non-sexual spore. Okay. And there is another one is called the Chytridomycota. They have a special spore that have flagella. So mostly this spore will be produced in the water ecosystem, aquatic ecosystem, because flagella usually used for swimming, okay, for movement. And then the last one will be all the fungi that do not produce sexual spore. So this fungi only produce a sexual spore. So mostly this one are yeast. Okay? Yeast are fungi that do not produce the sexual spores. They only produce the non-sexual spores. Okay? Sometimes this one is also known as imperfect fungi or deuteromycota. Okay, that's the scientific name. Imperfect fungi or deuteromycota. Examples are yeast. Okay, for identification, okay, if for example, for clinical identification, then take the sample and see under the microscope. Then we have to check what is the hypertype, whether septate or maybe non-septate hype. Okay, and then you see the spore, how it look like, whether it is sporangiospore, conidia, basidiospore, ascospore, or zygospore. Okay, so we perform this identification. And then colony texture, if it is mold, for example, what is the color? Maybe brownish, okay, yellowish, or maybe white color, and etc. Right? So this is the mold texture and pigmentation. Uh, Physiological characteristic, and also checking the DNA. Usually you may be using the PCR to see the genetic makeup of this fungi. So this is usually used for the methodology used for identification. Now, roles of fungi. Okay. Just remember, most of them are beneficial. Okay, they are actually what we call the primary decomposer. Okay, so together with bacteria, they are actually primary decomposer. It means that they decompose the waste organic. Okay, so like for example, if we throw our food outside, then it will be decomposed by this fungi and bacteria. It's become nutrient for plants. So they actually remove a lot of waste product, okay, especially organic waste product. They also, so this is the primary composer over here. They also produce good things like antibiotics. Some fungi like yeast is used for making alcohol, okay, making organic acid, vitamin, making food like bread, for example, okay, cheese, all right, yogurt. So these are beneficial roles of fungi, but some of them also have adverse impact, like they can cause disease. And again, disease that caused by fungi is called mycosis. They can also causing the allergy to some people, produce toxin, like aflatoxin, for example, that can kill uh, human. Okay, and then they also can destroy crops and food storage. Okay, so these are some roles of fungi. Okay, this is our focus in this class, which is mycosis, the infection that is caused by fungi. There are two types of infection. The first one is called the superficial infection, usually around the skin. Okay, so epidermis, this is a skin. This is the name of the infection. And then this is the name of fungi that causing this infection. We will learn this more or deeper in chapter 22 later. But right now it's just a list of some type of infection and location of the infection and the species or the fungi, the pathogenic fungi that causing this infection. Okay, again, the skin infection, tinea versicolor. This is caused by this fungi. And then in the hair, epidermis, and also maybe under the skin. Dermis is under the skin, like ringworms infection. Okay, so the infection under the skin, you're gonna see like this, like a ringworm. So this is caused by fungi. In the mucous membrane under the skin, okay, or in the nail, okay, like candidiasis. And this is caused by this fungi, okay, Candida albicans. Yeast infection maybe in the mouth, in the sexual organ, around sexual organ, okay, it's called the candidiasis. And then the second type of infection is called the systemic or deep. Systemic usually carried by blood and then going to organs inside our body, like for example lungs. Okay, lung infection. These are several types of lung infection. Again, we will learn this in chapter 22 later. Okay, so we're gonna learn more about this type of fungi infection or mycosis that enter the lung, lung and skin. Okay, so this like bacteria, I mean this fungi, for example, if it's in the skin, can cause skin infection. But if enter the lung, it can cause lung infection. Okay, I think we finished for this lecture about fungi. And then we will continue the next eukaryotic organism, which is protists, algae, and the protozoa. I'll see you later. Goodbye. Hi, everyone. So welcome to medical microbiology. So today we will continue chapter 5, survey of eukaryotic microorganism. We have lecture number 4 today, which is about protists. Okay, so we just finished lecture on fungi on the previous lecture. Now we will discuss about protists, which is algae and protozoa. So they are under the kingdom of protists. Okay, again, algae and protozoa. So algae, okay, they are many of them or most of them are photosynthetic organism, or they are autotrophic organism. Okay, remember they are able to make their own food. So auto means self, trophic means food. So they're able to make their own food. Therefore, most of the algae will be free living. They do not cause problem to human, except several algae, and they are called as a opportunistic parasite or opportunistic pathogen. They only cause disease when they enter into our body, maybe accidentally. For example, if we drink the contaminated water, or if we swim in the lake, drink the uh, lake water, and there are maybe some algae that can cause problem into our body, mostly GI tract problem. Okay, so again, algae mostly they are free living because they are able to make their own food. Now, different with protozoa, okay, some of them are free living, but some of them also can cause disease because these protozoa, remember, they are heterotrophic. Okay, so they have to get the nutrient from others. So they can enter our body and then they can cause problem. They become parasites inside our body. Example is malaria. Malaria is the parasite that caused by protozoa, and this is number one. Okay, number one infectious disease that causing death worldwide. Okay. Okay, we start with algae. Again, algae, it is photosynthetic organism. They able to make their own food. Therefore, they are also known as uh, autotrophic organism. They're able to make their own food because what? Because they have chloroplast, uh, chloroplast that are able to produce glucoses, okay, from uh, carbon dioxide and water. Remember the process of photosynthesis, which is water plus carbon dioxide, okay, and with the light energy or photo, then it will produce the glucose plus oxygen. Right? So this is the process of photosynthesis. This is the food. This is the food that is produced by this photosynthesis process. So this is happening, this process happening in the organelle is called chloroplast. So only organism that have chloroplast able to produce the food, which is algae and plant. Algae also have cell wall, just like plant, they have cell wall. Some algae actually have flagella. So this flagella is actually kind of animal characteristic, right? So that's the reason why, when we talk about protists, okay, so protist is a group organism that cannot be really tell whether it is animal or plant because they have both characteristic of plants and animal. So they usually divide it into algae and protozoa. Algae is a group of protists that have more plant characteristic. The protozoa is the group of protists that have more animal characteristic. Again, algae, mostly they are free living. They are free living in the fresh water or maybe ocean water. They call as a plankton. Okay, because they are plant, they also call as phytoplankton. Okay, phytoplankton is a group of algae. They are floating in the water. Okay, so this is a group of algae that may be causing disease to human. And again, mostly they are what we call the opportunistic pathogen. They can cause disease when they enter into our body by accident. Okay, like for example, if we drink polluted water with algae. Okay, this is the classification of algae. There are several class. Iglonophyta, okay, Pyrophyta, Chrysophyta, Pyophyta, Rhodophyta, and Chlorophyta. This is green algae, this is red algae, brown algae, this is euglenic algae, okay, this is dinoflagellate. So this is the organization, many of them unicellular. So for the euglenic or Euglenophyta, many of them one cell, okay, with flagella. Also, of course, it has chloroplast in it. Phyrophyta, many of them unicellular and also have flagella for movement. Chrysophyta, many of them unicellular, but some of them colony, filamentous colony that make a long filament. Phyophyta mostly multicellular, and you can see them usually on the ocean, like kelp and etc. So this is mostly microalgae. Okay, it's a big algae. Rhodophyta is the same. Many of them multicellular. This is red algae that they can use to make agar, okay, for food. Okay. And then chlorophyta, some of them unicellular, some of them filamentous or colony, and some of them multicellular. Okay. Protozoa, again, this one's gonna be the protist that has more animal characteristic. Therefore, many of them are heterotrophic organism. So it means that they have to get the nutrients from other organisms. Okay, some of them will be parasite because they are heterotrophic. Many of them is actually free living. Protozoa, they actually classified based on their motility advantages, whether they have flagella. If they have flagella, then it's going to be grouped in one division, whether they have cilia for movement or whether they have pseudopods for movement. Okay, so they're going to be classified into these different type of locomotion appendages. Okay, locomotor appendages. We will actually learn more in chapter 23 about algae and also protozoa. For the identification, okay, it's gonna be based on the shape, the size of the cell, type and number of locomotor structure, okay, like how many flagella, whether they have cilia as locomotor appendages, okay, number of nucleus, for example, okay, and then presence of special organelles on the cell, or maybe the ability to produce what we call the cyst. Okay, cyst is a structure that protects the animal. Okay, to protect the animal inside the cyst. especially it is made when the environment is not favorable for them. Okay, these are example of some pathogenic protozoa. Okay, again, we're gonna learn them or discuss them more in chapter 23 later. This is the first genus. It's called the Trypanosoma. They usually, okay, destroy the red blood cell. Okay, so they kill or destroy or eat the red blood cell. That's the reason why their primary disease or symptom or sign of this disease are anemia. Okay, because what? Because the red blood cells are being destroyed by this protozoa, the Trypanosoma. There are two main Trypanosoma that's actually separated in two different continents. Yeah, the first one is called the Trypanosoma brucei. This is in Africa, causing disease called the African sleeping sickness. And this is anemia, type of anemia. The second one is called the Trypanosoma cruzi. This is in American continent, South America. So it's called the disease called the Saga disease. And they usually need animal that is called as a factors. So the factor is mostly the type of insects. Okay, for example, for this Trypanosoma cruzi, the factor is called the kissing bugs. Okay, so this is the kissing bug, the factor. Why is called kissing bug? Because this bug is like to find the food around our mouth, especially when we sleep and we produce the drool. And this is the place where this animal like to absorb that. And sometimes they can bite our lips and then transfer this trypanosoma into our blood. And then finally the trypanosoma over here live inside our blood and can destroy the red blood cell. Okay, so again, it is transferred by blood sucking factor, like these kissing bugs. Okay, the second example of protozoa that can cause disease to human is called the ant amoeba. Okay, this is usually live in water. If we drink contaminated water with this ant amoeba, that it can cause disease called the ant amoeba histolytica. So this is in the GI tract. So this is amoebic dysentery. Okay, so this is a stomach ache problem, which is gastrointestinal disturbances. They can also enter into our blood system and then deliver into other organ like liver, lungs, or skin. So they can cause also some infection in these organs. Okay, I think we're done with the protists. Then we will meet again on the last lecture, which is helminths or parasitic worm. Okay, I see you later then. Bye. Hi, everyone. Welcome to Medical Microbiology. Today, we will continue chapter 5. This is going to be the last lecture. We will discuss the last example of eukaryotic microorganism, which is the parasitic worms or helminths. Again, we have discussed fungi, algae, and protists. Now we will discuss the helminths or parasitic worms. Parasitic worms, it is animals. So this is under kingdom of animals. Of course, they are gonna be heterotrophic. Okay, remember again, heterotrophic mean group of organisms that have to get nutrients from other organisms, whether eating it or absorb the nutrient from other organisms. Therefore, some of them become parasite, which is harm the host. Parasitic helminth, it is under the kingdom of Animalia, so they are actually animals. Of course, they are multicellular because this is a big type of microorganism. Okay, they have organ for reproduction. Okay, they have digestive system. They able to move and have a way to protect themselves. Okay? Many of them are parasite. They can stay in the tissue like under the skin or maybe in our intestine. They stay in our intestine. As many of them has a mouth part that can be used for attachment. So like a hook. So in their mouth they have hook that make them able to stay or attach in our organ like intestine. Okay? Some of them have sexual organ. Okay, so therefore they can be, some of them can be classified as male and female worms. They produce eggs. Okay? And then usually the fertilized eggs will go to the larva period inside or outside the body, the host body. There are two types of worms that can cause disease to human. The first one has a flat body. It's called the flatworms. And the second one, it has round body. Therefore, it's called the roundworms. Okay. Example of flatworm is tapeworms, like beef tapeworm or maybe pork tapeworms. Flukes, they usually can attack liver, this one, flukes. For the roundworm, there are several type, okay, like pinworm, for example. So this is example of roundworm that parasite to human. We will discuss this more in chapter 23 later. Okay, so in this chapter 5, this is just introduction of this type of helminths. Okay, again, the classification based on shape, the size, organ development, maybe sexual organs, okay, presence of hooks, sucker, okay, mode of reproductions, appearance of eggs and larvae. These are some structure or characteristic that usually used for worms identification. There are approximately about 50 species that are parasite to human. Okay, they distribute worldwide, but mostly in the tropical area. Okay, so mostly in the tropical area, they have more incidence of worm infection. They can also cause infection to animals like dog, for example. Okay, Usually, okay, they can be transferred from some mode of transportation, like from the food, eating the raw food, for example, the raw meat, for example. It can also cause the infections of the worm infection, or from the soil, drinking water, okay, from the soil or from the drinking water. They can also enter into our body through this transport of mode, okay, mode of transport. They can also carried by insect, okay, some insect factors. There are billions of humans that are actually affected by this worm infection. This is just an example of uh, cycle of pinworm, okay, which is the round type of worm that cause infection into human body. Like for example, this lady or this girl have infection in the skin, for example, and then it can cause self-infection. It can be transferred to her again, okay, from the site of disease, and then that worm can produce the eggs. The eggs can go maybe to the soil, for example, fertilize in the soil, and then when the kid play in the soil in the mud, it can be transferred into their bodies and then causing the infections on their body. So it can be, transferred to different type of mode, maybe from the soil, from the drinking water, from the food, okay, like raw meat, and etc. Okay, so this is just introduction of the parasitic worm. We will study them again in chapter 23 later. I think that's all for chapter 5. We will meet again uh, in chapter 6. Goodbye. Hi everyone, welcome to Medical Microbiology. Today we will discuss chapter 6, which is the introduction to viruses. So what are viruses? Viruses are non-cellular particle. So this is not living things. Okay, so this is just a particle with a definite size, shape, and chemical composition. This means that they are very specific, so we should be able to identify them based on this characteristic. So the observation of viruses started very early in the scientific development. Louis Pasteur, he postulated that rabies was caused by a living thing smaller than bacteria. Remember, Louis Pasteur was the scientist that postulated the germ theory, promoted the germ theory that infectious disease is caused by germs or microbes. So this is part of his germ theory. Dmitry Ivanovsky, okay, proposed the name for these small particles as virus, mean poison. And then Ivanovsky and Biering showed a disease in tobacco caused by virus. So this is the first observations of the disadvantage of viruses, which is it spoil the tobacco production. The virology itself was a multifaceted discipline. It means that many other disciplines like medical science, plant pathology, maybe science in animal science and animal pathology. They also learned about virus because there are many viruses that can cause disease to whether plant, animal, and even the virus can also cause infection to bacteria. There is no universal agreement on how and when viruses actually originated. Okay, and then virus, because they are very small, they are considered as the most abundant microbes on Earth, and they play important role in evolution of bacteria, archaea, and eukarya. Many scientists believe that virus is actually the bridge between non-living and living things. And viruses are obligate intracellular parasite. Okay, intracellular mean they have to stay inside the cell in order to reproduce. So therefore, they are parasite, which is causing harm to the host cell, and they only reproduce. Okay, obligate mean only or must inside the cell, stay inside the cell in order to reproduce. These are some properties of viruses. First, of course, viruses are obligate intracellular parasite. Okay, they can cause problems to the host cell, and they have to be inside the host cell in order to reproduce. The host can be any type of organism, starting from bacteria, protozoa, fungi, algae, plant, animal, including human. Okay? They are very, very small. They are ultra microscopic size. Their measurement is in nanometer, which is 10 to minus 9 meter. It's very, very small unit. Okay, so we cannot use light microscope to see virus. We need the electron microscope to see viruses. They are not cell, so it's not cellular. Okay? So they are very simple and very economical. Okay? So they only have two main molecules as their structure: capsid, the covering, and the nucleic acid in the center. They do not independently fulfill the characteristics of life, so they actually not living thing, but they are able to reproduce inside the cell. So they need living cell in order to fulfill their living characteristic. They are inactive outside the cell. Okay? So it's really easy to destroy the virus outside the cell, but they are very active inside the cell. It is going to be more difficult to control them when they are already inside the host cell. Basic structure only consists of the capsid. Okay, that's the first structure, and then the nucleic acid core. So two main components of virus. Okay, the capsid and nucleic acid core. The nucleic acid can be either DNA or RNA. It cannot be both. It has to be one of these. Okay, so that's the reason why there are two types of viruses based on this nucleic acid: DNA viruses and RNA viruses. The nucleic acid, the DNA, can be double-stranded. This is the most common type of DNA. So remember, it's double helix like this. So this is the most common type of DNA, okay, double-stranded. But there are some viruses that carry very rare single-stranded DNA. So this is rare. And the RNA, okay, the one that carry RNA, mostly will be single-stranded RNA, because RNA mostly just single strand. Okay? So single-stranded RNA. But there are some viruses that carry double-stranded RNA, which is again is going to be very rare. The molecules of the virus, okay, in fact, the high specificity is mean that the virus only causing problem or attached to a specific cell. So they causing problem to specific cell or specific tissue or organs. Like for example, hepatitis B viruses is only attached to liver cell and causing problem to liver cell. Flu viruses, they only attach to the respiratory tract mucosal cell and then causing problem to this respiratory tract. Okay? So this is respiratory tract infection. So they are very specific. Okay? And then they multiply by taking control of the host cell genetic material. They actually hijacking the host cell in order to reproduce. The lack of enzyme, so most of them do not have enzyme, therefore they need to be inside the cell in order to use the host cell enzyme in order to reproduce. They also lack of machinery, so they do not have ribosomes to make protein. So they have to be inside the cell using the host cell ribosome to make their own proteins. The size, again, is very, very small, okay, mostly in nanometer. The biggest virus may be this one, pox viruses. They are about 2 micrometer, but the rest of the virus over here will be very, very small in nanometer. This is maybe the smallest virus over here. It's very, very small. These are okay. These are bacteria. This is Staphylococcus aureus. This is E. coli. Okay, these are two bacteria. This is a coccus bacteria. This is bacillus bacteria. They are bigger, a lot bigger than virus. That's the reason why some virus actually able to kill these bacteria. They can cause infection and destruction to this bacterial cell. Okay, so virus can infect the bacteria. Okay, the structure again very simple. The main molecules are capsid and nucleic acid core, whether DNA or RNA. So that's the two main molecules or components of virus. Okay, so virus particle, the covering, central core. The main covering is capsid. So all virus will have capsid. Okay, so all virus have capsid. Some virus have envelope. Okay, so some have envelope, but not all. Then the nucleic acid, the central cause. Okay, all virus will have this nucleic acid molecule. Okay, whether DNA or RNA. Again, cannot be both. Okay, only DNA or RNA. And some, okay, some will carry enzyme. Not all. Okay, some virus has enzyme. Very rare, actually, the one that have enzyme. Again, we start with the capsid. Okay, subcapsid consists of protein. Is called the capsomers. So this is protein. Okay, so the capsid is actually proteins. And there are some viruses that have envelope. So outside the capsid, there is another membrane that cover the virus. This is what we call the envelope. The virus that have envelope, they also have spike. The spike is the attachment structure of the envelope viruses. And then virus that do not have envelope is called naked. Is naked viruses. The one that have envelope is called the envelope viruses. Okay, so two type of virus based on the outside layer. Okay, whether naked without envelope or envelope viruses. Now the capsid, there are two type of capsid, helical or icosahedral. So helical is, the helical is like this, and then icosahedral is mean that it's like crystal with 20 sides. So it's like a crystal structure. So that's the two type of capsid. So this is the helical type of capsid. So the DNA or RNA will be inside here, covered by this helical capsid. And then this is the icosahedral capsid. It has 20 sides. It's like a crystal, and then the DNA or RNA will be inside this protease structure. Now the envelope, again, remember not all viruses have envelope, but if they do have envelope, then they will also have spikes on it. Okay, spike is the attachment structure for the virus that have envelope. Most animal virus have envelope. So animal virus mean virus that attack animal cell, okay, including human cell. So humans consider as an animal. Therefore, the virus that attack human is considered as animal viruses. The functions of capsid and envelope, of course, for protection and also help to bind or attach to the host cell. They also assist for penetrations inside the cell. Okay, there are some non-typical viruses. Remember, typically virus has capsids and nucleic acid. Some of them also have envelope. Okay, naked viruses and envelope viruses. But there are some other viruses. Okay, in addition to envelope and capsid and nucleic acid, they have additional structure, like protein and lipid structure over here. This is actually the biggest viruses. This is pox viruses. They have that additional structure. This is non-typical. Another non-typical viruses is the one that attack the bacteria. It's called the bacteriophages. Bacterio mean bacteria, phages mean eater. So this is virus that eat or destroy bacteria. They have special structure over here. They have tail and the head. The tail is special structure that used for attachment. So they have the tail fibers that's used for attachment to the bacterial cell. This is the picture under the electron microscope of these bacteriophages. Because they look like a T letter, so therefore sometimes they also call as T phages. Okay, the T phages are the same as bacteriophages. Okay, the genome, the nucleic acid, again, either DNA or RNA, cannot be both. Therefore, based on this nucleic acid, there are two types of viruses, DNA viruses and RNA viruses. The number of genome that they carry is very varies. Okay, it means that there are some viruses maybe carry 100 of genes. There are some only carry 10 or 20 of genes. Okay, there are two types of nucleic acid again, DNA virus, DNA and RNA. Therefore, there are two types of virus based on this nucleic acid, DNA viruses and RNA viruses. Remember, mostly DNA are double-stranded. So mostly this DNA virus will be double-stranded DNA viruses. RNA viruses, mostly they're going to be single-stranded RNA viruses because mostly RNA are single-strand. There are two types of SS or single-strand RNA, positive or negative. Positive means they are able to synthesize protein directly using the ribosome of the host cell. So they're able to make their own capsid, they're able to make their own spike, for example, because capsid and spike are protein. So this positive sense RNA able to make the proteins directly. While the negative sense RNA, it has to be converted into positive sense in order to synthesize their protein. So basically, the positive sense RNA will reproduce faster compared to negative sense RNA. If we compare between the NA and RNA viruses, then the RNA viruses mostly will be more dangerous because they are able to reproduce right away. in the ribosome because they have the RNA and then they can make proteins right away. They can make capsids, spike. They can also multiply the RNA right away in the cytoplasm. Okay, so therefore they are more dangerous compared to the DNA viruses. DNA viruses, they have to enter into the nucleus in order to replicate. Then they will also produce RNA from their DNA inside the nucleus, and then the RNA will go out from the nucleus to synthesize protein. So the process is longer compared to the RNA viruses. It is the reason why most of the RNA viruses are more dangerous than the DNA viruses. They can destroy the host cell faster compared to the DNA viruses. Okay, so RNA viruses will be directly perform the synthesis in the cytoplasms. They do not need to enter to the nucleus. Okay, so they are faster to reproduce. Okay, the structure of nucleic acid. Okay, so some virus actually carry enzyme. So mostly they do not have enzyme, but there are some virus that carry enzyme. Example is HIV virus. So HIV virus carries specific enzyme. It's called the reverse transcriptase, which is the enzyme that able to synthesize DNA from RNA. Okay, so from RNA into DNA using this reverse transcriptase. Okay, basically, HIV virus is actually RNA viruses. So this is RNA viruses that able to create DNA, and this DNA will be stay in the host cell, the human host cell, which is the T helper cell. Okay, stay there. So whenever someone HIV positive will stay HIV positive because that DNA of virus is actually stay in the host cell. Okay? So this is what we call the reverse transcriptase. Okay? The common process, okay, is actually from DNA to RNA. So this is the common process of transcription. Okay, so DNA usually will be converted or transcribed into RNA using enzyme is called a transcriptase. So transcriptase will be used over here. This is the common process. So this is what happened in human cell. The DNA will be transcribed into RNA. Then the RNA will be used to make proteins. Right? However, HIV virus is actually have special enzyme that able to convert RNA into DNA, okay? And this DNA will stay inside the host cell. So whenever HIV positive, it will be HIV positive forever. Okay? Classification, because virus has specific size, specific characteristic, they actually can be also classified. But the classification will not follow the organism classification. Okay, they do not have domain, they do not have kingdom, they do not have classes, and etc. So you cannot put them in one of the three domains, them in one of the five kingdom of organism because they are not living organism. The classification start with the order, okay, usually end with Virales, so Caudovirales, Mononegavirales, Nidovirales. So there are three orders. And then under the order, there will be family, usually end with Viridae. And under the family, there will be genus and species. And it's easy name, they always finish with virus. Okay, so the name usually based on what type of the disease, and then just put virus on it. Okay, like for example, herpes. The virus that can cause herpes is going to be herpes virus. Sometimes we put also human herpes virus. So that's the name, simple name. Okay, the disease, and then the end with virus. Cycle, okay, the stages of viral reproduction. So if we talk about animal viruses, which is the virus that have envelope, okay? So most animal virus has envelope. And remember, if they have envelope, they also will have spike. Okay, so this is envelope viruses. It has 6 stages. Start with the absorption. The absorption using the spike. Okay, so the spikes will be used for absorption or attachment to the host cell. And spike is very specific for specific cell that they bind. Okay. Like for example, COVID-19 spike, they only attach to the cell that's located in, the mucus cell that's located in the nose or in the mouth. That's the reason why the primary transmission of COVID-19 is through the nose or through the mouth. Okay, because they have cells that have receptor for this COVID-19 spike. After absorption, there will be penetration. Okay, so where the virus will enter into the cell. Because they have envelope, then the whole virus will be enter into the cell. Therefore, they need and next stage, okay, which is uncoating. They need to uncoat or remove the capsid in order to release the DNA or RNA. So this is special. Okay, this is special stage for animal virus, which is it has process is called the uncoating. And after uncoating, the RNA or DNA will start synthesis. If it is DNA viruses, then the DNA, will enter into the nucleus and start synthesis inside the nucleus. If it is RNA, then the RNA will start the protein synthesis in the cytoplasm. Right? And then after synthesis, after they make capsid, they multiply the RNA, DNA, they make the spike, then they will assembly together. So this is the next process, assembly. After the assembly, then they will be exit the cell or release from the cell, mostly using process what we call the budding off or exocytosis. So when they budding off, let's say this is virus inside the cell. Okay, so let's say this is nucleus. They make capsid. Okay, they also multiply their RNA. This is synthesis, and then their assembly over here. The capsid will bind together. Okay, to make this structure to protect and cover the DNA or RNA side. And then they also put the spike on the host cell membrane. So when they're ready to exit, then the exit will process called the budding off, okay, or exocytosis. So this is going to be capsid with DNA or RNA. This is spike, and finally they will exit from the cell as a complete virus with envelope and spike over here. Okay, so this is what we call the exocytosis or bud off or budding off from the cell. So this is for animal viruses. Okay, so this is how the process start with the attachment, penetration, then uncoating, remember, and then this is synthesis of their DNA or RNA. This is synthesis of capsid. This is synthesis of spike. The spike then will go outside to the cell, okay, become part of the cell membrane, and then the capsid nucleic acid will assembly together to make this virus, and then finally it will budding off. When it is budding off, it take the part of the host cell membrane. So this virus over here is actually carry the host cell membrane that has spike on it. Okay, so the envelope is actually part of the host cell membrane. The absorption, as I mentioned before, the spike will absorb or attach to the specific cell that has specific receptor for this specific spike. So it's very, very specific. Like, for example, hepatitis virus only attached to the liver cell because only liver cell have receptor for the hepatitis viruses. Penetration and uncoating. So this penetrate the cell and then uncoating over here. And then replication, assembly, and then release. Okay, so this is assembly, start to the assembly over here, and then finally release by budding off. This is under the electron microscope. You see, several viruses actually exit from the cell. Sometimes when they exit, there are maybe 10,000 viruses will be exit from the cell in one exit, in one release. So the damage that can be caused by the virus is called the cytoplasmic effects. Maybe change the cell size and shape. Okay, and become bigger. Cytoplasmic inclusion. So when we see under the electron microscope, there will be some extra molecules inside the cytoplasm. And this is what we call the inclusion bodies or nuclear inclusion bodies. If it is DNA viruses, they will enter into the nucleus. Then there will be extra molecules, is inclusion bodies. Cell fuse. So some virus actually will attack one cell and then remove the cell membrane, and then the other cell will be also destroyed by the virus, destroy the cell membrane, and then two cells fuse together, become bigger cell. So virus able to cause some cell fuse together, maybe two cell, maybe three cell, four cell that fuse together finally become one big cell with many nucleus. Cell lysis is mean that virus actually, when it exit from the cell, it will destroy the cell. Like influenza or flu viruses, they perform the cell lysis. They destroy the cells of the host, the host cell. Some also alter DNA, like HIV viruses. They stay in human DNA inside the nucleus, and some transform cell into cancer. Remember, cancer is not infectious disease. This is caused by the cell that divided uncontrollably. Okay. However, some virus actually able to trigger this cancer cell. Like for example, hepatitis B, it can trigger liver cancer. There are two types of infection actually. The first one is called lytic infection or acute. Okay, lytic or acute. So acute infection is very fast. It destroys the cell within days. Okay? Like for example, Ebola, it can cause lytic or acute infection. It will destroy the cell within several days. Flu virus, for example, they also lytic type of infection. They destroy the some respiratory tract cell right away within days. But type of infection is called a persistent infection, maybe chronic or maybe latent. Okay, so chronic, persistent, or latent infection. The chronic persistent infection is mean that the sign of and symptom of the infection will be observed all day, all the time. Like for example, TB. Okay? Someone that have TB infection, you will be able to see the sign and symptom. Uh, like for example, he's coughing every night. He's gonna have a difficulty to breathe. For example, that's a chronic persistent infection. Sign and symptoms will be present all the time. While the other one is latent infection over here, it means that the sign and symptom only present during outbreak, during special time, like herpes, for example. So herpes virus, they are latent infection. They stay inside the human body, but only give the sign and symptom during outbreak time. Like for example, cold sore, that's a herpes virus. So you only have the sign and symptom of cold sore during certain time, maybe during winter time, so, for several days. After that, you will not have it again. You will not have it, and then you will have it again maybe next year, etc. That's what we call that latent infection. Again, some viruses are able to cause or stimulate cancer. This virus is called the oncoviruses. Onco mean cancer. Viruses mean virus that trigger the cancer. For example, papillomaviruses, human papillomavirus can trigger cervical cancer. Epstein-Barr virus can trigger the Burkitt's lymphoma cancer. Hepatitis B virus can trigger the liver cancer. Now, the second type of virus is the one that do not have envelope. Right? This is naked viruses. Example is the virus that destroy bacteria. That virus is called bacteriophages. They are naked viruses. Okay, bacteriophages. Mostly they will have what we call the lytic cycle, acute cycle, destroy the cell right away. Now, the stages will be the same, 6 stages, but there are differences over here. Okay, we start with the first one. Of course, it's going to be the same, adsorption. But remember, for the envelope virus, they use spike for absorption. This one, the bacteriophage will use the tail fiber, to attach to the bacterial cell. And then the second one is going to be the same, penetration. In this case, the DNA or RNA will be injected out from the virus into the bacterial cell. After that, replication right away. Okay? Remember, the replication is the same as synthesis. Okay? Remember, in the animal virus, in the envelope virus, there is uncoating first. There is uncoating. Number 3 is uncoating. But over here, they start the replication or synthesis right away. And then after the synthesis, of course, there will be assembly. And then this is the difference over here. Before they release, they need to be mature. So they have maturation. This is only presence in the bacteriophage or virus that do not have envelope. And then after that, they're going to be released. And when they release, they will destroy the cell. So mostly the release is a lysis. It's not budding off. It's not exocytosis, but it is lysis, destroying the cell. Okay, this is the process. Again, start with absorption. Okay, so absorption over here, and then penetration. Okay, so penetration. This is assembly. Synthesis or protein synthesis, and then this is assembly over here, and then maturation, and then release. Okay, when they release, they destroy the cell. You see the one, they actually kill the cell. This is what we call the lytic stage. But the virus, the bacteriophage, also able to go to the persistent infection. The name is not latent, it's not chronic, but the name is going to be lysogenic. For bacteriophage, the persistent infection is called the lysogenic, where they actually will not kill the bacteria, but they will put their DNA as a part of the bacterial DNA. So they're going to stay with the bacteria for a long time. Sometimes, when something happens, they can trigger this into this lytic process. Okay, so they can start, for example, with synthesis, assembly, and then release lytic over here. But they can also stay for longer time in this lysogenic condition. Okay, so again, the process is called the lysogeny, where the virus actually do not kill the bacteria, rather they stay with the bacteria for long term. Okay, so there are two again cycle, going to be acute or lytic cycle and lysogeny cycle. So this is acute is called lytic. Okay, the persistent is called the lysogeny. This is for bacteriophage. Now, the lysogeny sometimes can make the harmless bacteria become harmful. This is what we call the lysogenic conversion. Example, this one, we're going to use this, okay, Clostridium botulinum. When there is no infection of virus in this bacteria is actually harmless. But if this bacteria has viral infection in it, then this bacteria able to produce toxin is called botulinum toxin or Botox. And this is harmful to human, especially it will paralyze human cell, muscle cell. If it is the respiratory muscle, the muscle that used for breathing, then it will kill human, us, okay, because we are unable to breathe because of this Botox. Again, this Botox only produced by bacteria that have virus over here, okay, have virus that perform the lysogenic cycle, okay. There are some other examples also for this lysogenic conversion. Okay, so we compare between the bacteriophage, which is this is naked virus, with animal virus, which is it has envelope. This is envelope virus. Okay, so start with the attachment or absorption. It's gonna use tail, right? This is using tail fiber. And for envelope virus, using spike for absorption or attachment. And then after that penetration, usually injecting the DNA, RNA, this one will, the whole virus will enter into the cell. Therefore, after this, after the penetration for animal virus, there will be uncoating. Okay, and you don't see this uncoating in the bacteriophage. It's gonna start with the synthesis and then assembly right away. Okay, for the long-term infection for bacteriophage is called the lysogeny. But for animal virus is called latency or chronic, or sometimes it can cause cancer. Release from the host cell is going to be mostly immediate or lytic, right away lytic. But this one is going to be delay, sometimes immediate, like Ebola, for example. Sometimes it's delay, like HIV. Okay, so they usually with the process called budding off or exocytosis. Okay, so that's the difference between animal virus and bacteriophage. Remember, in the bacteriophage, before they release, they will have another process. It's called a maturation. Okay, so these are the difference between animal virus and bacteriophage. Animal virus has uncoating. The bacteriophage has maturation. How to culture the virus? Of course, because virus need living cell, then the culturing method will involve the living cell. Like for example, maybe culture the virus in the cell culture, okay, or in the embryo, like eggs, for example, or inside animal, maybe inside, put virus in the mouse, for example, or rabbit. Prion is smaller molecules than virus because it's only protein, but this protein can cause infectious disease to human. Eh? The infectious disease to human is called the Creutzfeldt-Jakob syndrome, or CGS. They can also cause problem to animal like cow. It's called the mad cow disease. So they actually attack mostly brain cell. So the same thing with over here, CGS is a brain cell damage. And prions are extremely resistant to the controlling agent like chemical or radiation, boiling. You cannot destroy prions with that. The other molecule that can cause disease to human is satellite viruses. This is a specific virus that need other virus in order to reproduce. So they need they will need to work together. So two viruses or three viruses, they combine or they work together in order to be able to replicate. The other one is called the viroid. This is just RNA, particle of RNA that can cause damage to other organisms. So far, viroids mostly identified in plant, so it's causing problem to plants, not animal yet. So this is the other type of molecules, similar or maybe smaller than virus, that also able to cause some damages to other organisms, including human. Uh, for treatment, uh, when it is outside the cell, virus actually easy to control. So you can wash your hands with the antiseptic soap. You can use alcohol, for example. It will destroy or remove the virus right away. And so it's going to be easy to control virus when it is outside the cell. However, when it is inside the cell, it's going to be more difficult. Because why? Because they already stay inside the cell. And so if we want to destroy them, usually we also have to destroy the host cell. So that's the reason why the antiviral drugs or medicine usually will cause serious side effects, because they actually causing problem to our cell. For example, uh, people with HIV positive, if they take the antiviral drugs for HIV, they usually have a lot of side effects like nausea, vomiting, feeling uncomfortable with their body, and etc. Because that antiviral drug is actually enter into the cell in order to attack the virus inside the cell, and it will cause, of course, some side effects that coming from the entrance of these viral drugs into the host cell. I think that's enough for chapter 6. So we finish with chapter 6. We will meet again with the next chapter, which is chapter 24. See you then. Goodbye. Hi everyone, welcome to Medical Microbiology. Today we will discuss chapter 24, which is DNA viruses. Before we go to that DNA viruses, we will discuss a little bit about types of infections that are caused by viruses. So just remember again that viruses are smallest particles or parasites with a very simple biological structure. It only consists of two main components, which is capsid and the nucleic acid core. The nucleic acid can be DNA or RNA. Okay, remember each virus only carry DNA or RNA as their nucleic acid. They cannot be both of them. Okay. The DNA viruses mostly double-stranded. Remember that because DNA, the main structure of DNA is actually double-stranded. So most viruses that carry DNA, they will carry the double-stranded DNA. But there are several viruses that also carry the single-stranded DNA. For the RNA, most viruses that carry RNA will be carry the single-stranded DNA. But there are some viruses that actually carry the double-stranded RNA. Remember again about virus infection. Okay, they are very specific to specific sites in our body. So it's only in particular site in our body. Like for example, hepatitis virus will only infect the liver cells, okay, because they are only limited to develop to reproduce in the liver cell. HIV viruses, okay, for example, they only attach and reproduce inside the T helper cell. Okay, so T cell is one of type of white blood cell in our body. Now, just remember also that most DNA viruses will have the effects inside the nucleus. Okay, they have to enter to the nucleus because remember in our cell, DNA are located in the nucleus. Therefore, DNA viruses will also send their DNA to enter to the nucleus in order to find or to use the enzymes. Okay, the enzyme that used for making DNA. Okay, the enzyme is the DNA polymerase. Okay, these enzymes is located inside the nucleus. Therefore, okay, DNA viruses will send their DNA into the host nucleus, okay, the host cell nucleus. RNA, they will multiply in the cytoplasm. Okay, remember RNA is used for making proteins. Okay, there are ribosomes in the cytoplasm that will make proteins from the RNA. So therefore, RNA viruses will reproduce in the cytoplasm. And again, because of that, the RNA viruses mostly will be more dangerous than DNA viruses because the RNA viruses will be ready to reproduce. They're gonna reproduce faster compared to the DNA viruses that have to enter into the nucleus first. The viral infection range from asymptomatic. Asymptomatic means does not give any sign and symptom, but the virus is actually inside our body, but there is no sign or symptom of this infection. Okay, so from asymptomatic, no sign, no symptom, to mild, okay, maybe mild symptom and sign like maybe flu-like symptom, fever, okay, and even some of them actually life-threatening. Okay, like for example, Ebola viruses, they can kill the patient, the host cell within 4-7 days. Okay, this is very, very dangerous type of viruses. Many viruses are strictly human in origin. Others are zoonosis. Zoonosis means it can infect animal and then from animal transfer to human. Okay, so for the zoonosis, mostly they will be carried by vectors. Okay, it will be transmitted by vectors. Vectors are animals, okay, that transfer virus from animal to human or from human to human. Mostly vectors are arthropod. Arthropod means the animal that have their legs on their chest, on their thorax. Example like insect, mosquito, flies, okay, or maybe like tick, uh, lice. Okay, these are factors, okay, animal factors that can transmit virus or infection from animal to human or from human to human. Okay, the viral infections, okay, sometimes okay, they start in the portal of entry. Remember when we talk about the portal of entry, like the nose, the mouth, okay, skin, okay, that's the portal of entry. So, so most viruses sometimes can cause infection and then destructions uh, on the cells or tissue in the portal of entry. Like for example, flu viruses, they actually destroy the mucosal cells on the respiratory tract, which is actually their portal of entry. Some virus, they can also enter into circulation, like blood circulation. Therefore, they sometimes can be transmitted through the blood, okay, to the blood. For example, blood transfusion, etc. Like for example, hepatitis B viruses, they actually reproduce inside the liver cell. However, they can carry it by the blood. So therefore, they are also present in the blood, but they do not reproduce in the blood. Okay, their reproduction place is in the liver, but they able to go to the blood circulation. Therefore, hepatitis B can be transmitted through blood. Okay, like for example, blood transfusion or maybe needle sharing. Okay, or injection. Uh, Common sign and symptom of viruses, okay, mostly rashes, fever, okay, muscle aches, respiratory involvement, swollen lymph nodes. This is the most common sign and symptom of viral infection. But again, for some virus, okay, like for example, hepatitis B, they can also have some other symptom in the liver because they actually destroy the liver cell, okay, because the liver cell destroyed, then they can have the sign like, for example, jaundice, because jaundice actually telling us there is a problem with the liver. The body defense, okay, so our body defense is combining the interferon, antibodies, okay, T cell, uh, and usually after viral infections, like for example chicken pox, and then after we able to remove, okay, or defend ourselves against this chicken pox, we usually have what we call the immunity, and mostly lifelong immunity. To someone that already have chicken pox, they usually will not get chicken pox again because they have the immunity against chicken pox. Many viral infections have what we call the lytic cycle. So lytic cycle is the same as acute, okay? Same as a acute infection. Okay, it's acute type of infection. So that's a lytic cycle. And some actually establish long-term persistent. Okay, so this is persistent. The top one is acute, lytic or acute. Example of lytic cycles is the flu viruses. So flu viruses actually destroy our cells very fast. Within maybe 7-10 days, okay, many of our cells will be destroyed by this influenza or flu viruses. But some other viruses, they establish long-term persistent infection. So this is what we call the persistent infection. There are two types of persistent infection. The first one is called the chronic, okay, chronic infection. And the second one is called the latent infection. Now, the chronic infection, yeah, is the infection, is a persistent infection, long-term infection, where the sign and symptom usually present all the time. Okay, like for example, hepatitis B. This is a long-term persistent infection. Then usually people that have the hepatitis B will have the sign and symptom of this disease. Okay, AIDS, for example, it's HIV infection, AIDS. Okay, this is a chronic infection because what? Because the patient that have AIDS then will have the disease, the sign and symptom with them all the time. Okay, so that's what we call the chronic infection. The latent infection, okay, this is a type of persistent infection that only give the sign and symptom during outbreak. So most of the time, the virus is actually in a dormant condition, in the dormant stage. So they stay inside our body, but they will not give the sign and symptom of disease until the outbreak time. Example is herpes, okay, like oral herpes, for example. Usually, this oral herpes virus will stay in our nervous system. It will only give the sign and symptom, like for example, you can see the sign of symptom is oral herpes uh, on the mouth, okay, next to the mouth, okay, at the skin of the mouth, okay. Then it is only usually present during certain time, what we call the outbreak time. The same thing with the herpes simplex number 2 or genital herpes. They stay in our nervous system dormant for a year, for example, and then during outbreak time, then they will give sign and symptom around the skin of the genital area. Okay, that's what we call the latent infection. Okay, that's the difference between chronic infection and latent infection. Again, make sure you do understand that there are two types of viral infection. The first one is called the acute or lytic cycle, and this is usually very fast. Okay, they will destroy the cell very fast. Example is flu viruses, Ebola viruses. There's also having a lytic cycle. They destroy the cell in a very fast action. And then the second type of infection is called the persistent infection, which is long-term infection. There are two types of persistent infection, chronic and latent. Chronic means long-term infection where the signs and symptoms are present all the time. And then the latent infection means the persistent infection or long-term infection where the virus mostly in dormant stage only give the sign and symptom of disease during outbreak time. Okay. Okay, this is also illustrations of the type of infection. This one is a lytic infection over here, okay, where the virus actually out from the cell and then destroy the cell. Okay? This is latent infection, mostly stay in dormant stages, and then in certain time, like outbreak time, then they can give the sign and symptom. Okay, this is a chronic infection. When the virus actually reproduce and then kill the cell slowly for a long time, and they able to reproduce and release the virus from the infected cell. Example is hepatitis B, for example, or AIDS or HIV infection. It's a chronic infection. This is just another type of kind of viral infection when the virus actually stay and causing changing in our cell. So like, for example, maybe causing the cancer or tumor. Okay? Some persistent viruses are oncogenic. Oncogenic means it trigger cancer. Okay, oncoming cancer. So there are some viruses that actually able to cause or to trigger cancer. Example is again hepatitis B. Hepatitis B, this is infect the liver cell, but also trigger the liver cancer. Some virus can cross the placenta and then causing permanent damage in the embryo. And this is what we call the teratogenic viruses. Okay, terato means embryo. So teratogenic viruses is the virus that can damage the embryo. Oncogenic viruses is the virus that can trigger cancer. Okay. The viral infection, there are several ways. Like for example, take the sample, isolate the samples, and then put it in the animal or living tissue culture, and then perform serological tests with the antibodies or some other tests with the gene, like PCR test, for example. I think that's all for these introductions about the viral infection. On the next lecture, we will talk about DNA viruses that cause infection to human. See you. Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 24, the envelope DNA viruses. So basically, we're gonna talk about DNA viruses that can cause disease to human. Okay, just remember that human is considered as animal. Okay, therefore, the virus that infect human is called the animal viruses. Okay, so animal viruses are viruses that infect animals, including human. You just remember that the animal viruses, okay, mostly they will have envelope. Okay, so animal viruses mostly have envelope, and they also have spike. Okay, remember the virus that have envelope, usually they have spikes as the attachment structure. Okay, so therefore, okay, in this chapter, when we talk about the animal viruses or human viruses, then mostly the one that will infect human will be the envelope DNA viruses. Okay, so envelope DNA viruses. Okay, now remember that most DNA, okay, the usual structure of DNA is double helix, which is double stranded structure. Okay, therefore, mostly the one that infect human will be envelope double stranded viruses. Okay, this is going to be the most common type of DNA virus that infect human, which is they have envelope and they carry double stranded DNA. There are some other type which is naked. Okay, naked is doesn't have the envelope. dsDNA, double stranded DNA, that also can infect human. Okay, again, the most common one will be envelope double-stranded DNA. There are also some naked double-stranded DNA that infect human. And very rare over here, okay, there are actually also naked single-stranded DNA that can infect human, but it's gonna be very, very rare, maybe only one type of naked single-stranded DNA. Okay, I'm gonna make it bigger over here. Okay, so DNA viruses. Okay, yeah, remember, because human is animal, so this is gonna be animal viruses, as mostly animal viruses will have envelope. Okay, so it's gonna be on this left side, which is the most common type of animal viruses. It has envelope. Now, because this is DNA viruses, okay, remember the most common type of DNA is double-stranded. So therefore, okay, over here on the left side will be double-stranded DNA. So when we combine this, okay, envelope and double-stranded DNA, so these ones will be the most common type of DNA virus that infect human will be envelope double-stranded DNA viruses. Okay, so will be envelope double-stranded DNA viruses. Right? There are three groups, okay, of these envelope double-stranded DNA viruses, which is pox viruses, okay, pox viruses, herpes viruses, and hepatinaviruses. This is actually another name for this hepatinaviruses is hepatitis B virus. Okay, so these are the three groups of envelope DS, okay, double-stranded DNA viruses that infect human. And this is the one that we will discuss in this lecture. Okay? The other one or the next type going to be naked or non-envelope. Okay, so this is going to be naked virus. Okay. Double-stranded. Okay, naked double-stranded viruses will be three groups: adenoviruses, polyomaviruses and papillomaviruses. And then the very rare is going to be naked single-stranded DNA, which is only one type, parvoviruses. And this group over here, the naked viruses, will be discussed on the next lecture. So we will focus in this main group first, the envelope, the envelope double-stranded DNA viruses. Okay, this is just comparison over here, just a list of all the DNA viruses that can cause disease in human. The first one is smallpox, which is pox viruses. This is another pox viruses. So these two over here, pox viruses. Okay, and then herpes viruses, there are 8 types. Okay, 1, 2, and this is number 3, number 4. Actually, this one number 4, Epstein-Barr, number 4, CMV. CMV is actually number 5, herpes viruses. Okay, all of these herpes viruses. And number 6, 7, there is actually one more herpes virus, number 8. Right? So there are going to be 8 herpes viruses. And the last one for envelope double-stranded DNA will be hepatitis B viruses. Okay, so this one still the first group, which is the double-stranded envelope double-stranded DNA viruses. Adenoviruses, human papillomaviruses, papillomaviruses. This is going to be the next group. Okay, this is the naked double-stranded DNA. And the last one over here, this is the only one. This is naked single-stranded DNA virus. Okay, so we start with the first group, which is the envelope. Okay, DS, okay, double-stranded DNA viruses. Okay, the envelope double-stranded DNA viruses. And remember, there are 3 groups: pox viruses, herpes viruses, and hepatitis B virus. We start with the first one, which is the pox viruses. Okay, the first group, pox viruses. Okay, so the pox virus is actually the largest type of virus, okay, in size. So they are the biggest type of virus because they have additional structure in addition of their envelope, the capsid, and nucleic acid. So they have other structure outside that make this virus become the biggest type of virus. So if you see over here, they have envelope inside. Okay, outside the envelope there is some additional structure over here, and then the inside the DNA. Okay, so for the pox viruses, the first ones will be the one that causing the small pox. The name of the virus is the variola. There are 2 types of variola. Variola major, which is very, very dangerous viruses. It has about 80% mortality. It means that 8 of 10 people that have this virus will die because of these viruses. It's very, very dangerous viruses. The other one is variola minor, which is not that dangerous, mostly just give mild infection. But variola major was the most dangerous one of this smallpox type. And the good thing that this disease actually has been eliminated. It has been eliminated by vaccination. So vaccination worldwide is actually able to eliminate these smallpox viruses. It's considered as the virus that has been eliminated from this world. Okay, it's I think it's around 1980, 1970 when the vaccination worldwide were started. And this is what happened if someone have the variola major infection. You see the smallpox infection over here. It gives a lot of skin swellings, and this is going to be burned, okay, feeling itchy. And if, remember, this is 80% mortality, it means that 8 of 10 people that infected by this virus will die. If they able to recover from this disease, they usually will have scars on their skin. So the scars will stay on their skin. Okay, so this is smallpox. Again, it has been eliminated due to the vaccination effort worldwide. The vaccination, okay, so this is actually the first vaccination worldwide using species of virus is called vaccinia. So vaccinia is the name of virus that's very similar with the smallpox viruses. This is actually kind of cowpox viruses. Right? So vaccinia is the virus that used as a vaccine for smallpox. This is the reason why the name is called vaccine. Okay, vaccination. This is actually taken from the first vaccine that was created for worldwide vaccination. Okay, so vaccinia viruses is used as vaccine to vaccinate people against smallpox disease. It started around 1970, and then around 1980 or 1990, you know, the whole world actually has been vaccinated. Yeah. So this vaccine is still continue in order to protect people from the recurrence of this disease. And this is how the vaccination done. Usually after the vaccination, they will have, if you get vaccination of this, you might have this scar on your shoulder, on your skin, in the place where you get the vaccination. I believe young people will do not have this vaccination right now. Another pox viruses that also can infect human is the molluscum contagiosum. The name is telling you this is contagious. And this is usually causing the infection around the genital areas. Therefore, this one is also can be transferred through the STD, as sexual. So this is sexual transmitted disease. Okay, another pox viruses that can infect human is monkeypox and cowpox. Monkeypox is more dangerous or more contagious compared to the cowpox. Okay, so many people actually have monkeypox infection due to their handling monkey. Maybe they work at the zoo, or people that have a monkey as their pets, for example, they might get this monkeypox infection. Right? Cowpox is considered as rare, causing infection in human. If they do, it's only on certain skin area. It's not spread around or the whole body. Okay, thus we finish with the second group, the pox viruses. Now we go to the next group of enveloped double-stranded DNA, which is the herpes viruses. Okay, herpes viruses. Okay, it is also known as human herpes viruses. Okay, or HHV, human herpes viruses. There are 8 of them. Okay, 1 until 8. Okay, 8HV viruses. And there are 1 until 8 herpes viruses. Most of these herpes viruses is actually latent. Remember, this is one of the persistent infection where the virus stay inside the patient body, the host cell, and then will give the sign and symptom during the outbreak time. So mostly this virus will be dormant inside the patient body and only give the sign and symptom of disease during the outbreak time. Okay, this is how it look like. You see it has envelope, right? Because this is envelope viruses. Okay, and then on the envelope, they also have the spike. Spike is used for attachment. And then capsid will be inside. Okay, this is icosahedral type of capsid. And in the middle, there will be double-stranded DNA. Okay, so this is the structure of herpes viruses with envelope, spikes, capsid, and then double-stranded DNA in the core, in the inside. Again, there are 8 type of herpes viruses, okay, human herpes viruses. The first one and 2 change the name into herpes simplex 1 and 2. So it used to be human herpes 1 and human herpes 2. Now it's become herpes simplex 1 and herpes simplex 2. Right? So this is the one that usually causing the infections around the facial area for the herpes simplex 1 or genital area for the herpes simplex 2. And then human herpes virus number 3 changed the name into varicella zoster viruses. Varicella is chickenpox. So this is the one that causes chickenpox on children. And then when they growing into adult, then the adult will have what we call the shingle. It is caused by the zoster viruses. Okay, varicella zoster viruses are the one that causing chickenpox on children and shingle on adults. And then number 4 is the EBV, okay, human herpes virus number 4 become EBV, which is Epstein-Barr viruses. This is the one that causing disease called the mononucleosis or mono disease or kissing disease, because they are transmitted from the mouth. Okay, the infection usually around the salivary glands, which is in the mouth. Okay, and then number 5, human herpes virus number 5, it becomes CMV, cytomegaloviruses, mostly non-symptomatic, except for some people, maybe transplant people or AIDS people, they might causing the meningitis, for example. Okay, so CMV can cause meningitis for some patient with lower immune system. And then number 6 and number 7, they are very similar. They can cause disease called the roseola or baby skin rash. So skin rash on baby is called the roseola. This is caused by human herpes virus number 6 and 7. And then the last one, human herpes virus number 8. This is usually for 8 people, it can cause skin cancer. It's called the Kaposi sarcoma. Okay, so again, there are 8 types of human herpes viruses, 1 until 8. Number 1 and 2 change into herpes simplex viruses. Number 3 become varicella zoster viruses. Number 4 become Epstein-Barr viruses. Number 5 become cytomegalovirus. And number 6, 7, and 8 stay at human herpes viruses number 6, 7, and 8. Yeah. So human simplex virus number 1, or human herpes virus number 1, this is the one that causing the cold sore. Okay, so usually the infections around the facial area. And then for number 2, human simplex virus number 2, usually causing infection around the genitalia. Okay, so this is the tables that's showing us the difference between human simplex virus 1 and 2. Mostly the name of the infection that caused by a human simplex virus number 1 is around the facial area, like herpes labialis, okay, in the lips, ocular herpes around the eyes, okay, gingivostomatitis and pharyngitis, it is inside the mouth, in the pharynx, and in the around the teeth, okay. And for the herpes simplex virus number 2, mostly the infection is around the genital area. Okay. Transmission with close contacts. Okay. For the HSV number 2, okay, this is going to be close contacts and sexuals, because they are located around the genital area. Latency, dormancy. So the herpes simplex virus number 1, they stay dormant in the nervous system. In the place is called the trigeminal cranial nerve. Okay, if you learned ANP2, okay, human anatomy and physiology, you learned about there are 10 cranial nerves. Then cranial nerve number 5 is called the trigeminal ganglion or trigeminal cranial nerve. Is the one that supply the face. Okay, therefore they stay in this area and then will give the sign and symptom during the outbreak times around the facial area. Okay, and then for the human simplex virus number 2, they stay in the nervous system around the sacral ganglia. Okay, sacral is below our back, so in the bottom area. Okay, so that's the reason why their infections mostly on the genital area or in the buttock area. Okay, so genital area, buttock area, thigh. This is the place where they can give the sign and symptom of infection. While for human herpes simplex virus number 1, it is mostly on the face, on the mouth, okay, near the eyes, etc. Now, some complication. This virus can also causing infection in other area. Like for example, if it is in the hand and the finger, it's called the herpetic whifflow. And if it is attack the brain, it's called the encephalitis. Okay. Okay, again, we mentioned before that herpes simplex number 1, they stay and they enter the number 5 cranial nerve, okay, which is trigeminal cranial nerve, and stay there, dormant there, and then give the infection or the sign and symptom of disease during the outbreak time, mostly around the facial area. And then herpes simplex number 2, they will stay in the lumbosacral spinal nerve. Okay, this is a nervous system on the back. Okay? And therefore, when they give the sign and symptom during outbreak time, mostly around the genital area, where the place where it's close to this spinal nerve. Okay, this is showing us where the herpes simplex virus number 1 stay dormant, which is here. Okay, so this is the cranial nerve number 5 is called the trigeminal nerve, and this cranial nerve supply the face, okay, facial area. So this is the one that, for example, sense what happens in the skin around our area, and this is also the nerve that control the muscles in our face. Okay, like for example, when you talk, when you move your uh, facial muscle, giving the facial expression like laughing or talking, smile. Yeah, it's actually controlled by this trigeminal nerve, and the herpes simplex virus number 1 dormant or stay in this cranial nerve. Okay, therefore, it will give the sign and symptom during outbreak time around this facial area. Okay. Again, for type 1, herpes simplex virus type 1, this is some infections or disease that can be caused by this virus, herpetic gingivitis, especially in the young children. This is inflammation in the mouth, okay, gum, tongue, soft palate, and lips. And it can also give disease called the herpetic keratitis, which is ocular herpes. This is around the eyes, okay? Like for example, maybe conjunctivitis, okay? Sensitivity to light. This is also some sign and symptom of ocular herpes. This is just showing us the oral herpes, okay, in the mouth, in the lips, in the tongue, and maybe also on the gums. The type 2, okay, herpes simplex virus type 2, this is the one that causing what we call the genital herpes. Okay, it's going to be sign and symptoms will be present around the genital area. Okay, this is some problem that is caused by this herpes simplex number 2. Inflammation of urethra or urethritis, painful urination for girl or woman, cervicitis, okay, inflammation of cervix. Okay, this is example of skin lesions and the genital area caused by this type 2 herpes simplex virus. Okay. For the newborn, the herpes simplex number 1 or number 2, sometimes it is fatal. It is dangerous for them, okay, because baby usually they have a very limited immune system. Their immune system is not really or fully developed yet. So therefore, infection caused by this virus can be fatal for them. Now, the prevention usually, if the mother have the herpes simplex virus, for example, if she has the SSV number 2, then the doctor usually will give the suggestion that the mother to deliver the baby with the C-section, okay, in order to protect the baby from getting infection by the HSV number 2 that mostly infect the genital area. This is what happens if the baby have that infection. HSV number 2, you see that skin lesion the whole body, and it's going to be very painful. Sometimes it's become fatal for this newborn. Okay, so for the miscellaneous herpes infections, we mentioned before herpetic with low. Okay, this is infections on the hand or fingers. It's a local infection, or it might enter into the brain, causing what we call the encephalitis. Okay, inflammations of the brain tissue. This is the picture of herpetic with low, okay, infection of fingers because of the herpes simplex virus. Treatment, okay, usually using drugs, and usually the drugs will end with vir, okay, acyclovir, famciclovir, valacyclovir. This is telling us this drug usually used for herpes infection, okay, herpes virus infection. Okay, we done with the herpes virus number 1 and 2. Now we go to the herpes, human herpes virus number 3, which is now become varicella zoster viruses, or called the VZV3 viruses. Now varicella is the one that can cause chickenpox, usually on children. And then when the children growing up, if they have chickenpox, then mostly they will also have shingles on their older time. Okay, when they're adult, so adults usually will get shingle when they have chickenpox during their children time. Okay, so varicella causing the chickenpox, the zoster causing the shingle. This is actually the same virus. They stay in patient nervous system, okay, and then causing chickenpox during children time and zoster during adult time. This is chickenpox, and this is where they stay, which is in our nervous system. So stay in our nervous system, dormant in our nervous system. Sometimes, like stress, can trigger this virus to give sign and symptom in our skin. So example is this one over here, shingles. So shingles is caused by the zoster viruses, which is varicella viruses also. Okay, I think we're gonna pass this one. The treatments, I'm gonna be using the same drugs, okay, like acyclovir, famciclovir, because this drug is used mostly for herpes viruses. Okay, so VZV is also type of human herpes viruses. This is type number 3. Okay, this is type number 4, human herpes virus number 4. It's become Epstein-Barr viruses, or EBV, and this is known as the virus that can cause monodisease or mononucleosis, or sometimes this disease is also known as kissing disease. Because what? Because this is mostly transferred during by kissing, because this virus actually stay in the salivary glands, which is in the mouth. Okay. And the disease called the mononucleosis or monodisease. And this is the problem with this monodisease, because the incubation time is very long, about 30 to 50 days. So about 2 months, the incubation time. It means that that person carry the disease, but he does not have the sign and symptom for about 2 months. So it means that he has the disease, he carry the disease, he carry the virus, and able to transfer to other people without he realized that he has that virus with him. Okay, so this is usually how this disease transfer from people that have the virus but do not have the sign and symptom, okay, which is during the incubation time. And then when the virus gives the sign and symptom, then usually these people will be very weak, fever, flu-like symptom. They usually will stay at home because they're getting sick now. But before that, for 50 days, they actually did not realize that they had the disease and spread it to other people. And this is just showing us the sign of disease in the throat. Okay. And this disease also, okay, this virus, the EBV number 4, can also trigger a cancer. It's called the Burkitt lymphoma cancer, which is the cancer on the salivary gland. So this EBV virus is actually also known as the oncogenic virus. Okay, remember oncogenic virus is the virus that can cause or can trigger the tumor or cancer. They also can trigger the other type of cancer, which is nasopharyngeal carcinoma. So this is another type of cancer that can be triggered by the Epstein-Barr virus, which is human herpes virus number 4. This is the picture of a child with Burkitt lymphoma, which is tumor or cancer on his salivary glands. Okay. Diagnosis, remember, you can identify virus, bacteria using whether phenotypic, genotypic, or immunologic methods. Okay, so this is the serological method, is a genotypic method to identify the presence of the viruses, okay, using the antigens, antibody reaction. And then treatment, also very similar with other herpes viruses, using drug like acyclovir, or maybe also injection of monoclonal antibodies. This is the sign of someone that have this Epstein-Barr viruses. This is phenotypic, if you know. Phenotypic means observe what happens with that patient. Maybe take a blood sample and then compare with the the normal blood and the abnormal blood. Okay, so this is a phenotypic type of diagnosis or method. So comparing the normal lymphocyte, for example, over here, with the lymphocyte of people that has the Epstein-Barr viruses, okay, in its blood. Okay, we go to the next human herpes virus, which is human herpes virus number 5. The name changed into CMP, cytomegaloviruses. Usually this virus give no symptom for many people, so mostly asymptomatic, even though that person maybe carry that viruses. Sometimes or most of the time there is no symptom. However, for some other people, maybe 8 patients, okay, remember 8 patient is the persons that have lower immune system. Therefore, this virus can cause problem to them. Okay. This is the sign. Okay. It's actually taken from the lung cell. And you see the someone that have this virus will have a bigger lung cell. So this is abnormal lung cell because of that virus. This is normal lung cell over here, which is smaller. Okay, so this virus can be transmitted through the saliva, respiratory mucus, or milk. This is from mother to the baby, urine, semen, and cervical secretion. Okay. For someone that just have a transplantation, organ transplantation, usually if they have this virus, it can cause some of this disease like pneumonitis, hepatitis, myocarditis, or meningitis. So this is this virus, okay, actually can trigger this disease for someone that have lower immune system like AIDS people or someone that just get the organ transplantation. The treatment almost similar with other herpes viruses, which is using the drug for herpes viruses. Again, usually end with vir, ganciclovir, valganciclovir. This is examples of drug that used to treat the CMV viruses. Okay, and then the last one's gonna be 6, 7, and 8. So this is human herpes virus 6, 7, and 8. So they keep the name, okay? Human herpes virus number 6, number 7, and number 8. For number 6 and number 7, they are very similar, causing similar disease. It's called the roseola, which is also known as baby diaper rash. Okay, so baby so baby diaper rashes. This is a roseola. This is how it looks like. So you see the skin of this baby has a lot of rashes, and this is mostly caused by number 6 and number 7. For number 8, okay, the number 8 over here, for 8 patients, they usually causing tumor, skin tumor or cancer is called a Kaposi sarcoma. So this Kaposi sarcoma is usually caused or triggered by human herpes virus number 8. Okay, now the last one for envelope double-stranded DNA that infect human is the virus that can cause hepatitis B. So hepatitis B over here. This is the sign and symptom of hepatitis, which is one of this yellowish of the skin color. So this is hepatitis B viruses. The name of virus is called the HEPATNA viruses. HEPA means liver, DNA viruses. So you combine, become HEPATNA viruses. This is the only DNA viruses that can cause hepatitis. The rest of hepatitis is actually caused by RNA viruses, like hepatitis A, C, E, and D. They are caused by RNA. We will talk this on chapter 25. Okay. This is just comparison between 3 hepatitis, A, B, and C. C and B is very similar. It can be transferred through the blood transfusion. That can cause also liver cancer. Whether the A usually transferred to the oral or fecal contamination. Okay. Okay, mostly hepatitis B and C, they are chronic. It means that they stay in the liver cell and then causing destruction to the liver cell, and even they able to trigger the liver cell become cancer or tumor. So that's for B and C, while A usually will not causing the liver cancer. So basically, B and C are more dangerous than A, hepatitis A. Okay, and this is the Hepatna viruses, Hepar DNA viruses. Okay, this is the one that causing the hepatitis B. Okay, so this is uh, hepatitis B virus. Okay, so the Hepatna viruses is the one that causing the hepatitis B disease. Now, hepatitis B is disease on the liver, and it's special in the liver cell because that virus has the receptor on the liver cell. So remember, this is gonna be, this is envelope viruses, right? Capsid, DNA, envelope with spike. Okay, so this spike is gonna be very specific for, let's say this is liver cell. Okay. Okay, so this spike will have the receptor over here. Okay, receptor for hepatitis B viruses. So only liver cell have the receptor for hepatitis B viruses. Therefore, this virus only attached to the liver cell and causing destruction, even can trigger the liver cancer. And after they reproduce in the liver cell, they actually able to go or shed out into the blood. And so the blood will carry that virus, but there will be no reproduction in the blood. Yeah? They're only present in the blood. Therefore, that can be transmitted from one person to another person, maybe through the blood transfusion, sexual intercourse, and some other blood related transmission. And so that's the reason why this hepatitis B can be transferred through the blood. Because what? Because they are actually present in the blood after they reproduce in the liver cell. So they can be transferred to other people. Okay, so again, this virus will reach the liver cell and then multiply, reproduce on that liver cell. Okay. This is some sign of symptom of people with the hepatitis B infection. Like for example, jaundice, which is yellowish of the skin of the skin, and also yellowish of the white of the eyes, fever, okay, low white blood cell count, arthritis, some rash around the the chest area, enlarged liver because of this infection. This is some of the sign and symptom of hepatitis B infection. So for the diagnosis, mostly using the immunologic test like ELISA or maybe RIA, radioimmunoassay, or maybe can use genetical, genotype type of method of identification. Okay, and I think we're done with the envelope double-stranded DNA. Then we will go to the next lecture, which is... Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 24, DNA viruses that infect human. Today will be the last lecture for this chapter. We will discuss the non-envelope or naked DNA viruses. Okay, so remember again this chart. Okay, DNA viruses that infect human. Okay, remember, because human is animal, therefore this virus will be considered as animal viruses. Viruses that infect animal, including the human. And remember again, most of the animal viruses has envelope. Right? Therefore, the envelope virus will be here. And then because this is DNA viruses, then most of the DNA viruses will be double strand. Okay, so it's double helix like this. Okay, so it's going to be double strand. Okay, therefore, the most common type of DNA viruses that infect human will be envelope double-stranded DNA viruses, which is this side over here. Okay, there are three groups. Okay, fox viruses, herpes viruses, and hepatna viruses, which is this is hepatitis B virus. Okay, now we already finished with this envelope double-stranded DNA on the previous lecture. Now, in this lecture, we will talk about the second type of DNA viruses, which is the non-envelope or naked viruses. Okay, so it could be carry the dsDNA or double-stranded DNA, or maybe carry single-stranded DNA or ssDNA. For the double-stranded DNA, so naked viruses that carry double-stranded DNA will be three groups, which is adenoviruses, polyoma viruses, and papilloma viruses. The naked single-stranded DNA will be only one, which is parvoviruses. Okay, remember, this is over here, the naked single-stranded DNA will be the least common type of DNA virus that infect human. Okay, again, the most common type will be it has envelope and carry double-stranded DNA. Okay, so we will continue with the double-stranded DNA, so naked, non-envelope, double-stranded DNA. Okay, will be three groups. So this one, the adenoviruses, papillomaviruses, and polyomaviruses. So this is naked, naked dsDNA. Okay, this one carry double-stranded DNA. The parvovirus will be naked, carry single-stranded DNA. Okay, only one, which is parvoviruses. Okay, we start with number one, the adenoviruses. Again, this one will be naked viruses. Okay, so this is non-envelope viruses or naked viruses that carry double-stranded DNA. This is under, this picture is under the electron microscope, how it look like. It's just capsid over here. Okay, so this is actually capsid, icosahedral capsid, right? So there is no envelope and no spikes on these viruses. So mostly the disease is related to respiratory tract infection. Okay, like it's similar to colds, pharyngitis, and maybe also when it is infecting the eyes, it can cause conjunctivitis, okay, and keratoconjunctivitis, which is infections around the eyes, okay, in the eyes. Okay, the second and the third one, papilloma and polyoma viruses. They are very small, non-enveloped dsDNA, eh, causing some persistent infection, and sometimes it can cause tumor, eh, cancer and tumor. Okay, so papillomaviruses is also known as HPV, human papillomaviruses. This is the one that infect human. And by the way, if you reach age 16 or 18, I think it is required to get this HPV vaccination, yeah, to try to prevent from cancer, especially for women. Try to prevent from the cervical cancer that is sometimes it is triggered by these human papilloma viruses. The most common type of infection is called the common warts, which is a type of skin infection. Right? And this wart can recur. It means that they actually stay the virus dormant inside our body, and it can recur after after maybe we able to remove these common warts from our skin. Okay, there are two types of warts. The first one is called the common seed wart. This is on the skin, okay, skin of the body, in our hand, maybe our leg. But the other one over here is called the plantar wart. This is a little bit bigger and painful, because mostly if you have this, it's going to be difficult to walk or to wear shoes, for example. This one also can cause what we call the genital wart, and this is one of the most common STD, sexual transmitted disease, in the United States. Okay, so this is just the common seed wart on our skin, and this is the genital wart. Okay, again, as I mentioned before, the human papillomaviruses can trigger cancer. Therefore, usually young adults are required to have the human papillomavirus vaccination. So this is the vaccinations that some of you maybe have to take it, and usually that vaccination is a double vaccination. You will get first vaccination, and then there will be a second booster vaccination, okay, for this human papilloma viruses vaccination. Okay, this virus can also cause a disease called JC and BK viruses. This JC and BK is actually the abbreviation of the patient, the first patient that have this kind of viral infection, okay, this kind of disease, JC and BK viruses. That's for the polyoma viruses. Now we go to the last one, which is the non-enveloped ssDNA. So it's gonna be capsid, and then the single-stranded DNA inside the capsid, okay, in the nuclear, in the center, okay. Mostly this virus causing infection to animal like cats and dogs is called a distemper. Okay, so mostly on the cats and dog, but it can also causing infection to human. The one that causing infection to the human is called the B9 viruses. Okay, so this is the parvoviruses type B9 that causing the infection is called the erythema infectiosum, or the most common name for this disease is called the fifth disease, usually on children, which is rash on the skin, and mostly the rash is present on the cheek. So the cheek of this child will be reddish, just like, you know, someone just slapped his or her cheek. So this is a cheek rash on the children, and it's also known as the fifth disease, and this is caused by the adenoviruses. There is another virus that has to be associated with these adenoviruses. It's called the adeno-associated viruses, and this is a defective virus that cannot replicate without the real adenoviruses. So this is what we call the, remember, the satellite viruses, the virus that need another virus in order to replicate. Okay, I think that's all for chapter 24, the human DNA viruses. And again, just remember, the most common one will be the envelope double-stranded DNA viruses. There are three groups, okay, the pox viruses, herpes viruses, and hepatna viruses. And the second one will be naked double-stranded DNA. Okay, and the last one, which is the least common one, will be naked single-stranded DNA virus. That's all for chapter 24. And we will see you again on chapter 25, which is RNA viruses that infect human. Goodbye. Hi, everyone. Welcome to Medical Microbiology. So today we will discuss chapter 25, which is RNA viruses that infect human. On the first lecture, we will discuss the first group, which is the segmented single-stranded RNA viruses. Okay, compared to the DNA viruses, the RNA viruses that infect human is actually more diverse. It means that there are more type of RNA viruses that infect human compared to the DNA viruses that infect human. So these are the list of several RNA viruses that infect human. The most common one, as we know, so this is the one that causing infection almost every year, which is the influenza viruses. There is another viruses called the hantaviruses, parainfluenza, mumps viruses, morbilliviruses, respiratory syncytial virus, rabies, SARS, this including the COVID-19, this is coronaviruses, Ruby viruses, Hep C viruses, St. Louis encephalitis virus, yellow fever, dengue fever, including Zika fever over here. And then HIV, the one that causing AIDS, polio, the one that causing poliomyelitis, Hep A, rhinoviruses, norovirus, rotavirus, Ebola. And the last discussion will be about the prions, which is just a proteins. It's not really virus, but is unfolded protein that cause infection to human, especially in human brain. Okay, now we go to the type of RNA viruses that infect human. It is really similar with the classification of DNA viruses. Remember, human is considered as animal. Therefore, this virus will be considered as animal viruses, which is what it will mostly have the envelope. Animal virus mostly have envelope. So it has capsid, and then the envelope, and of course there will be spike on the envelope that will be used for attachment. And inside the capsid there will be the nucleic acid. In this case will be RNA. So RNA. So these the envelope viruses. And just remember, because mostly the RNA is single-stranded, therefore the left side over here, the most common type will be the single-stranded RNA, ssRNA. Okay, and some of the RNA is actually segmented. Segmented means more than one segment. So like this one over here, this one has 2 segments. RNA. Non-segmented is the one that only carry 1 segment of RNA. So if we compare, it's gonna be look like this. So this one, the non-segmented, will only have 1 single-stranded RNA, and the other one over here, the segmented, maybe 2, maybe 4, maybe sometime 8. There are 8 segments. Okay, so this is segmented, and the other one is non-segmented. So this is segmented. This is non-segmented single-stranded RNA. Okay. So again, this will be, okay, the left side over here will be the most common type of RNA viruses that infect human, which is the one that have envelope, but including this HIV over here, the one that have envelope and carry single-stranded RNA. The other one over here will be the one that do not have envelope. This is the naked viruses, and the most common of these naked viruses will be the one that carry single-stranded RNA. So this is ssRNA, and the least common one will be the one that carry double-stranded RNA. Okay, so the this one gonna be like this, double-stranded RNA, and this one will be just single-stranded RNA. No envelope, because this was this naked naked viruses or non-envelope viruses. Okay, so for lecture 1, chapter 25, we will focus in this one first, okay, which is the envelope, segmented single-stranded RNA. There are three groups, okay, flu viruses or orthomyxoviruses, and then Bunia viruses and arena viruses. So this is going to be our discussion, our topic for lecture 1, chapter 25. Okay, so it's going to be the envelope segmented single-stranded RNA viruses. So if we make the picture, it's going to be this capsid, then this envelope, spike, and the envelope for attachment, and the single-stranded RNA. There are more than one segment. Okay, so that's going to be how the structure of this virus, okay, going to be envelope segmented single-stranded RNA. Again, the first one, flu viruses or orthomyxoviruses, Bunia viruses, and the arena viruses. We start with the first one, which is the orthomyxoviruses, or the flu or influenza viruses. This is the most common viruses that attack human. Every year there will be flu, there will be influenza, and every year there will be flu shot, different flu shot. Because what? Because this influenza virus, they actually can change their spike, and they also can substitute their RNA segment. Therefore, almost every year there will be different subtype of influenza. So the flu shot that we have last year maybe will not be effective for this year. The one that we have this year, maybe it will not, really effective for the next year, because the flu is always changing. But it is better to have the flu shot every year, in order to protect us from the flu that may be spread during that year. Now, the most common one and the most dangerous one is type A, type A flu viruses. And this type A actually have different subtype based on the spike. So the spike, which is the H spike and the N spike. So the spike for the virus, the flu virus, it has two subtype, which is H spike, okay, and the N spike. Now the H spike is actually, there are H1 until H15. So there are 15 subtype of H spike. The N spike, it has 9 types, so 1 until 9. That's the reason why it has number over here. So H1N1 is mean that it has subtype 1 for the H and subtype 1 for N1. And this is considered as the most dangerous type of flu viruses. And this is the one that killed millions of people in 1918, 1919, which is 100 years ago. It's called the Spanish flu. And then it's recurrent 76, 77, 91, and 2009. So this is the most dangerous type of flu viruses, flu A. And then H2N2, it's appear mostly in Asian pandemic, Southeast Asia, and then in China, H3N2 in 68 and 94 in Africa. So these are the type of A viruses that's considered very dangerous to human. And then B and C, they are less virulent or less dangerous compared to the type A flu viruses. Now, how they infect human? And remember, there are 6 stages of cycle of viruses. And because this is animal viruses, then there will be spike, and spike will be used for attachment. So this is going to be the first stage, which is attachment. Okay, the spike will attach to the respiratory tract cell. Okay? So therefore, the most common site for infection will be the nose. Okay, because nose is part of the respiratory system, respiratory tract. Okay? So attachment to the cell on the respiratory tract organs. And then after that, the second stage will be the penetration. And the RNA of viruses will be released into the cytoplasm. There are 8 segments. Remember, this is segmented RNA. So there are 8 segments of RNA that are carried by flu viruses. And these 8 actually is negative sense. Remember the difference between negative sense and positive sense of RNA. The negative sense cannot be used for making protein. Therefore, this negative sense of RNA will be entering into the nucleus of the host cell. Okay, inside the nucleus, this negative sense will be converted into positive sense. So this one is positive sense. It will go out from nucleus into the cytoplasm. And then they will use our ribosome, the host ribosome, to make the spike. So the spike then will be attached into the host cell membrane, into our cell membrane of the respiratory tract cell. And then inside the nucleus, the positive sense over here will be also used to replicate into negative sense of these RNA viruses, the RNA segment over here. And then this RNA segment will be out, million of them actually, and then every 8 of them will assembly. So this is the next step going to be assembly, okay? And then after that, the last step will be release from the cell. When it is releasing from the cell, it will carry the cell membrane of the host cell and the spike over here. So therefore, the envelope, remember, the envelope of animal virus is actually part of the cell membrane of the host cell. And flu virus is causing what we call the lytic or acute cycle or infection. It means that they actually kill our respiratory tract cell very fast. Within 7, 5, 10 days, they actually destroy a lot of our respiratory tract cell. And therefore, if our immune system unable to take care of this infection, then mostly, the patient will be die. That's what happened with the Spanish flu that killed millions of people, because of the destruction of respiratory tract cell by these flu viruses. So this is a lytic or acute type of infection, really fast type of infection. Usually, within 10 days, if our immune system able to defend our system against this virus, then we'll be cured, after 10 or 14 days of sickness or illness or flu. And again, the key of the influenza or flu infection is this spike over here. So the spike. And there are 2 types of spike, age spike, and there are 15 of age subtype, 1 until 15. And then the N spike, there are 9 subtypes, so 1 until 9. Okay, so therefore, usually the flu virus sometimes will be named based on what type of spike that they carry. So like H1, N1, H2, N2, H1, N5, H2, N9 maybe. Okay, therefore there will be a lot of type of flu virus actually, just based on the spike that they carry. And this spike, and let's see over here, this is the H spike, the blue one, and this is the N spike, the brownish one. So they have different structure, and they can be changed. So maybe this is H1, this is N1. Maybe this is H2, this is N5, for example. Okay, so they're able to change this spike every year. There's the reason why the flu shot for this year will not be effective for the next year. The flu shot that we have last year maybe will not be effective for this year. Therefore, we have to have or to get flu shot every year because of this changings or drift of the spike every year. Okay, so again, that's what we call the mutation. So this virus, the flu virus, is actually mutated every year. They can do what we call the antigenic or antigenic drift, which is changing the H, the spike, the H and N spike over here. They change it. They can change it. So therefore, it's going to be different virus this year compared to last year, and then there will be maybe next year will be different type of virus just based on the spike. This is what we call the antigenic drift. The second mutation is called the antigenic shift. It means that they actually able to shift or change the RNA from one virus to another virus. For example, here, this is a bird flu viruses. This is the avian viruses, which is the one that attack bird, and this is human viruses. There are two viruses over here, bird viruses and human flu viruses. And then they actually able to infect the same animal. Let's say this is swine. Okay, and then inside this swine, then what happened? They able to change maybe one or two segment. Like for example, the blue segment is coming from the bird, and the red segment is from human. And then just one blue enter into the human viruses. What happened? It's become new viruses compared to these viruses. This is already different type of viruses. Therefore, if, for example, we already immune against these viruses, because we have disease or we have flu shot for these viruses, and on the next year, maybe this is the virus that infect human. What happened? Then we will not have the immunity against these new viruses over here. Then we will be ill. We will have the sickness from this type of viruses. So just remember, the flu viruses, they change, they mutate every year, very, very fast. Again, there are three main strains, and the most dangerous one or the most virulent one is type A, and this is the one that have killed a lot of people lately. So especially older people, they are have a higher risk of getting this flu A infection. Therefore, it is required for older people to get the flu shot every year in order to protect them against this influenza A viruses. Influenza B and C, this is considered as less virulent or less dangerous compared to type A. Again, type A is highly acute. Remember, this is acute type of infection and highly contagious respiratory infection. This is seasonal and pandemic. It means that it can be transferred across the states, across the countries. We know the sign and symptom for these viruses. So diagnosis using some immunologics tests, treatment mostly using some flu drugs, okay, like maybe Tamiflu for H1N1, okay, Relenza, and vaccine is usually available every year. Okay, the second type of segmented ssRNA envelope ssRNA virus is Bunya viruses, and this is a kind of endemic. It means that's a local infection. It's not really pandemic. Several types like hantavirus and Sin-Nombre viruses is considered as the most dangerous type of these Bunya viruses, and usually it's transmitted by insect, like insect, like a mosquito and ticks. The last one will be the arena viruses. This is also mostly pandemic. I mean, endemic in South America, like Argentina, Bolivia. It can cause hemorrhagic fever. Hemorrhagic fever means the patient will have a high body temperature and sometimes with bleeding, accompanied with bleeding. So it's kind of type of dangerous viruses also. I think that's all for the first lecture, the segmented SS single-stranded RNA viruses. On the next lecture, we will discuss the non-segmented single-stranded RNA. Goodbye now. Hi everyone, welcome to Medical Microbiology. Today we will continue chapter 25, RNA viruses that infect humans. On the previous lecture, we will talk about the segmented single-stranded RNA viruses. So in this second lecture, we will talk about the non-segmented single-stranded RNA viruses. So it's going to be structure is going to be like this. So this is envelope viruses, right? Envelope with spike, okay, and capsid in the middle. And because this is non-segmented, it means that it's only one segment of single-stranded RNA in the center. So this is the illustration of this envelope, non-segmented single-stranded RNA. There are 6 groups in this non-segmented type of RNA, which is the first one is paramyxoviruses, and then rhabdoviruses, coronaviruses, togaviruses, flaviviruses, and filoviruses. Now we start with the first type of envelope, non-segmented RNA, single-stranded RNA viruses, which is myxoviruses. So this group has many subtypes. The first one is paramyxovirus that can cause parainfluenza and mumps, which is swelling on the parotid gland, parotid salivary glands, under the cheek. So swelling of the cheek. And then morbilliviruses, this is the one that causing the most contagious type of disease, which is measles. So this is the most contagious type of disease, measles. And then pneumoviruses, the third type, is the one that causing the cell to be combined together to make a giant cell. It's called the syncytium, a giant cell that has a lot of nucleus, because this virus actually break the cell membrane of one cell and then combine it with another cell, and finally it will make a giant cell with many nucleus on it. So the first one is parainfluenza. This parainfluenza is caused by the paramyxovirus. And this parainfluenza is very similar with the influenza, but with less contagious and less dangerous compared to the influenza that's causing by automyxo. Okay, remember on the previous lecture we talked about the influenza, which is influenza A, B, or C. So this one is called the parainfluenza, which is less contagious and less dangerous compared to the influenza. Mostly in children, so the parainfluenza usually attack children. And this is the sign and symptoms, you know, minor cold, bronchitis, bronchopneumonia. The second one is the virus that, you know, is the same virus actually. Paramyxovirus is causing disease, what we call the mumps or parotitis, inflammation of parotid gland over here. So under the cheek, you have a pair of salivary gland. It's called the parotid salivary gland. And this virus actually infect this parotid gland and then causing the swollen on this area. This is what we call the parotitis or mumps disease. Okay, and usually there is a vaccination for this mumps and measles. Is called the MMR, mumps, measles, and rubella. So this is MMR vaccine. Usually children, they will have this vaccination in order to protect them against measles, the mumps, and rubella. Okay, measles. Again, this is the most contagious type of disease. Okay, so this is a very, very contagious. The ID, infectious dose for this measles is 1. It means that one virus will be able to cause or start disease to the human, to the host. And human is the only reservoir for these measles viruses. And this measles virus is actually morbilli virus. This is the one that causing the measles. And there are 2 types of measles. The first one is called measles, and the other one is German measles. The first one is the most contagious one. It's caused by the morbillivirus. The second one is not that contagious. It's caused by rubiviruses. This is some of sign of measles. Okay, so you can see the spot. Okay, rashes in the mouth is called the Koplik spot. This is the very significant or specific sign for measles, and also maybe some rashes on the skin, mostly in on baby or children. Again, measles is the most contagious type of disease, and there is a vaccination for this, which is MMR. So mumps, measles, and rubella vaccine. The last one for this virus, so viruses, it is called the pneumoviruses. And from the name, this virus is actually infect the lungs and causing the lung, some of the lung cell become giant cell. It's called the syncytium or syncytial cell, where many cells combine together because of this virus become a giant cell that has many nucleus. Okay, so multi-nucleate cell, a giant cell that have a lot of nucleus in it. This is some of the symptoms: fever, rhinitis, pharyngitis, otitis, and also croup. And this is mostly on children and baby. Now the second one, the second type of virus is the one that causing rabies. The virus is called the rhabdoviruses. So this is the second type of non-segmented single-stranded RNA, which is rhabdoviruses causing the rabies. Rhabdo mean bullet. It's like a bullet, and this is under the microscope. This is the illustration of that structure and the envelope, and then you see the spike over here. Okay, and then the non-segmented SSN will be inside this center, long single-stranded RNA. Okay, rabies mostly transferred through the wild animal bites. So the reservoir is actually in wild animal, mostly mammals, like maybe dogs, okay, bats, okay, this type of wild animal that carry the rhabdoviruses and can cause rabies to human. And if the human have that virus, for example, get bit by the wild animals, then there will be 4 stages of that disease. The first stage will be called the prodromal phase. Start with a fever, nausea, vomiting, headache. And then the second stage after that is going to be furious phase, which is start with the agitation, disorientation, and mostly they will be, the patient will be afraid of water, hydrophobia. And then after that, the next dumb phase, the patient may be paralyzed for a while, disoriented, and even if it is not healthy, even causing death to the patient. So these are the 4 stages of rabies. This is the distribution of rabies in the United States. It is actually based on the distribution of the animals, the wild mammal that carry that disease. Okay, in the northern area and middle area, mostly carried by skunk. So skunk is the main reservoir and resource for the rabies in the middle part of United States. In the southern part, mostly caused by fox, fox that carry the rabies, rhabdoviruses. And the western part is also skunk. And the eastern part, mostly carried by raccoon. Raccoon is the source and also reservoir for rabies in the eastern part of the United States. This is some of diagnosis and also preventions and management of rabies. Okay, so treatment mostly using passive and active post-exposure immunization. Controlled by vaccinations of domestic animals. So like maybe dog, for example, they should get vaccinations against these rabies viruses. Okay, the next ones, number 3 will be coronaviruses. It's called coronaviruses because this virus look like a flower. So corona is actually mean kind of flowers. It's very nice structure of this virus under the microscope. Okay, this virus mostly attack the human respiratory system. Okay? So like a common cold, but it can also causing some very dangerous infection like SARS, okay, severe acute respiratory syndrome. This is in 2001, 2002. This become pandemic. And Middle East Respiratory Syndrome or MERS. This is mostly pandemic in the Middle East area around 2012. And then the COVID-19. This is the one that's still causing pandemic in some countries like India right now. Okay, so but in the United States, I think it's kind of lower down. Okay, it's kind of decrease the infection of this COVID-19, and mostly this is airborne transmission. And they actually mostly, you know, the spike, if this is the virus, okay, so the spike mostly attached to the receptor in the respiratory cell, like in the mouth, in the nose. So therefore the primary transmission of this virus will be through the nose and mouth. Okay, so again, this is the sum of severe type of disease that caused by the coronaviruses. SARS in 1912, and then MERS in 2012, and this is the sign and symptom of this disease, mostly on the respiratory tract infection. And the problem is if it is actually attack the lung, it can cause pneumonia and problem with breathing. This is the newly type of disease that caused by the coronavirus in 2019, and still in some area, and even in the United States, there are still some thousands of people that still have this type of infection, which is COVID-19. This is the name of disease, which is coronavirus disease 19. So this is the year when that disease is started. Okay, so coronavirus disease 2019. Okay, the next one will be rubella. It's caused by rubiviruses, also known as German measles. So it's very similar with the measles, but you know, it's less dangerous. And then also vaccinations, it's going to be the same like vaccination that used for measles, mumps, and this one, rubella. So the name of the vaccine is MMR, okay, which is mumps, measles, and rubella vaccine. So it's protect against 3 type of disease, which is mumps, measles, and rubella. Rubella is also known as a German measles. Okay, there are 2 types of German measles. The first one is called the postnatal rubella. This is after the birth. Okay, so mostly kids and children will have this kind of symptom. And the second one is called the congenital rubella, which is mostly affecting the baby or the fetus, the embryo inside the mother wombs. It can cause miscarriages. Okay. The next one, which is flaviviruses, can cause disease called the hepatitis C. So this is the name of the virus. Okay, this is the 5th type of non-segmented single-stranded RNA viruses, which is flaviviruses. can cause disease called the hepatitis C. And this hepatitis C will be very similar with the hepatitis B. Okay, can be transferred with the blood transfusion, for example. Okay. So it's blood contacts. Okay, like blood transfusion, needle sharing, or maybe blood transfusion. Okay. I think should be one more over here. I don't think we have. So this is number 5. Okay, the flaviviruses is number 5. I think I forget to put one more, which is the filoviruses. Yeah? And this is the one that causing the very dangerous type of disease, which is Ebola. And Ebola transfer through the liquid, blood liquid that is produced by the patient. So usually, the patients will die within 5 to 7 days after infection. So this is a type of lytic. Okay, so remember lytic or acute infection, where the virus actually destroy the cell very, very fast. And this destruction will cause bleeding. So therefore it's called a hemorrhagic fever type of infection. So it can cause fever and then bleeding, and then finally it will kill the patient within 5 to 7 days. So it is very, very dangerous and also contagious type of disease. Okay, I think that's all for lecture 2. Okay, for chapter 25, which is non-segmented type of RNA. It should be not end yet, so this is wrong over here. So the next lecture, okay, I forget to change this one. The next lecture will be HIV, which is very special virus that causes AIDS. Okay, so lecture 3, chapter 25 will be about HIV viruses. Okay, I'll see you then on the next lecture. Goodbye. Hi everyone, welcome to medical microbiology. Today we will continue chapter 25, lecture 3, which is about the HIV virus. Okay, I think you still remember this grouping. So remember that RNA viruses that infect human is considered as animal viruses. Mostly they will have envelope, and then because this is RNA, mostly they will be single-stranded RNA. Okay, so this is the left group over here is the most common type of RNA virus that infect human, which is enveloped with single-stranded RNA. And there are 3 types over here, which is the first one is segmented, envelope segmented single-stranded RNA. So if it is envelope, there will be spike. Always remember, envelope virus, usually they have spike for attachment, and then capsid, which is proteins, and the single-stranded RNA. Because this is segmented, it's going to be more than one segment of single-stranded RNA. Okay. The second group will be envelope non-segmented single-stranded RNA. Say it is envelope. Then remember, gonna be spike. Okay, and it's gonna be capsid. And because this is non-segmented, then there is only one segment of single-stranded RNA inside. Now, the next one over here, the one that we will discuss in this lecture, which is HIV or retroviruses. One of them is HIV. If you see the structure, it's actually like this. It has envelope, of course, spike, okay, and then the capsid, and it's actually carry two segments of RNA. Okay, now based on the structure, then these retroviruses or HIV is actually considered as segmented type of viruses. However, it is become special because this virus carry special thing, which is enzyme. So enzyme over here. Okay, I'm gonna change with black color. Okay. Enzyme carry special enzyme is called the reverse transcriptase. So this is the one that make these retroviruses or HIV become special, and it is put in its own group, which is retroviruses. So this is the one that we'll discuss today. Okay, again, HIV infection and AIDS. So the type of retroviruses, the one that carry special enzyme, which is reverse transcriptase, that infect human is called the human immunodeficiency viruses or HIV. Okay. And this is a type of acquired immunodeficiency. There are two types of immunodeficiency actually. The first one is called the primary immunodeficiency, which is it is the patient have it since they were born. The acquired one is the one that is caused by something, like for example, is caused by these viruses. So this is acquired, which is after the patient, the person born, then they get the disease, and the disease causing them to have less immune system, right? Causing them to be deficiency on their immune system. So this is a type of acquired or secondary. This is another name for acquired, which is secondary immunodeficiency syndrome or disease. It is first emerged in early 1980. Okay. This is some of sign of symptom of AIDS, which is severe pneumonia caused by pneumocystis xerophaeae. Okay, ordinarily this pneumocystis xerophaeae is actually harmless. So this is not really causing problem for healthy, normal people. Right? So this is actually fungi. But if this fungi infect people with HIV, with AIDS, then it causes them to have a very severe pneumonia. Okay? They also will have some vascular cancer. It's called the Kaposi sarcoma cancer. You can see this sign of this cancer on the skin. So their skin become blackish color because of this cancer, Kaposi sarcoma. They also mostly will have sudden weight loss, swollen lymph nodes, and of course, they will have general loss of immune function. So basically, they are unable to fight against infection. So AIDS patients, they usually die not because of the HIV, but mostly because of the secondary infection. The infection that usually not that severe for normal people, but for people with AIDS, it can kill them. Okay. 1959 is the year when the first AIDS was documented. Now the structure, and as I mentioned before, this virus actually carry two segments of RNA. So this is actually segmented envelope single-stranded RNA. So you see the envelope, the capsid over here, and then the two segment of RNA plus there are several enzymes that this virus carry. Because just remember, enzyme mostly, mostly virus do not have enzyme because they're gonna use the enzyme of the host cell. So this is a special for this virus because they carry special enzyme. And there are several of them. Okay, retrovirus, the genus of lentivirus. This is actually for the HIV under the genus of lentiviruses. Again, they have two single-stranded RNA. So this is double single-stranded RNA. So they carry two segments. And this is the problem with this virus. Okay, this virus carry the reverse transcriptase enzyme. It will make the DNA, okay, their DNA from their RNA. And this DNA then will be integrated with the host DNA and stay with the host DNA forever. So it means that whenever someone, okay, diagnosed positive for HIV, then they will be positive HIV forever because this DNA of this virus, okay, is actually integrated with the host cell. Okay? And of course, it causes disease called the acquired immunodeficiency syndrome or AIDS. Okay, this is what is special with the HIV. They have special spike. Okay? So spike that only infect the host cell that has special receptor for that spike. So it has a receptor for the HIV virus spikes, and only CD4 cell is actually have that receptor. So what is CD4? CD4 is the cell that become T helper. Okay, so remember there are several type of white blood cell. Okay, one of them is called the T cell. Okay, there are B cell, there are T cell, there are neutrophil, eosinophils, monocyte, and etc. And one of them, one of white blood cell types is T cell. And there are two types of T cell. Usually, the first one is called the TC or cytotoxic C, and the second one is called the TH or T helper. And T helper is the one that will be infected by the HIV because only T helper that have receptor for HIV spikes. So it looks like this. So this is HIV virus with the spike. This is the spike. And that spike only bind to this receptor, GP120 receptor, which is only located in the T helper cell. So that's the reason why the HIV is actually only infecting the T helper, and they will kill the T helper cell little by little. So if we have some million of T helper, it will be killed little by little by this HIV until it is, their concentration gonna be very low. And then what happened after it is very low? That patient become AIDS. So it needs several, sometimes months or years incubation period before that patient become AIDS. Okay, from HIV positive into AIDS, it takes sometimes months or year. Okay, transmission mostly through the sexual intercourse, and that's the main type of transmission. Also through the transfer of blood. Okay? Babies can be also infected before or during birth, okay, from the breast feeding and also from the umbilical cord. Okay. Okay, so this is the transmissions of HIV through the blood, like for example, injection, transfusion, or maybe a broken skin, and then that person, for example, acquire blood that has HIV on it, and then this HIV will enter into his system. Okay? So usually, yeah. The route of transmission through the blood and also sexual intercourse or transmission. Okay, again, first noticeable this disease in United States is 1980, 1984. And this is the sixth most common cause of death among young people in the United States. And men accounted for about 75% of new infection. IV drug abuser, they consider as a carrier for this disease. Can be spread to the sexual, especially heterosexual and homosexual intercourse. In 2009, the number of infected individuals worldwide is estimated to be 35 million, and about 1.2 million in the US. The most populated area with AIDS patient is in the sub-Sahara continent in the Africa country. Okay, this is the transmission. This is taken from CDC data. Okay, the higher transmission is through the male-to-male sexual contact, which is homosexual, about 67%. Okay, and then heterosexual contact, female about 16% and male about 7%. Injection or drug use about 3% transmission for female, and for male about 4%. And male-to-male sexual contact and IDU, okay, about 3%. So from this, then we should be able to notice that the homosexual actually have a higher risk of getting the HIV infection. Okay. The reason is because during that intercourse, there may be some broken skin that can cause that virus to enter to the inside the body easily. This is just showing us the rates of HIV infections among adults in 2014 in the United States. As we see, usually bigger city, they have higher infection rate. Okay, again, the HIV will enter through the mucous membranes under the skin, and finally they will reach the blood. When they're reaching the blood, then they will be able to attach to the T helper, T helper cell, and then inside the T helper, they will make their own DNA from their RNA, and this DNA then will be combined with the DNA of human T helper cell. Okay, this is maybe easier to understand over here. First, when the virus enter the blood, then what happened? They will find the T helper, okay, the T helper, which is one type of T cell, and this T helper has a special receptor for the spike, okay, of these HIV viruses. So this is the first stage is attachment. Okay, remember again, stages of virus life cycle start with the attachment, and then after that is penetration. Remember, this is animal viruses, then they will penetrate with the capsid, and therefore after penetration there will be uncoating over here, right? So uncoating, where the capsid is open, and the RNA, remember there are two RNA that this virus carry, will be released into the cytoplasm, plus there will be release of this enzyme into the cytoplasm. And this enzyme then will be used to create DNA from the RNA. So this DNA is actually produced from the RNA using the reverse transcriptase enzyme. Okay, now just for your information, this is actually the normal productions or the normal process of DNA, RNA in animal cell or in the cell. Okay, remember if it is the cell, okay, the cell membrane, this is the nucleus, for example. Okay, inside nucleus there will be DNA, okay, which is the gene, and this DNA will be actually converted into RNA. That's the normal process, RNA. And this RNA will be out from the cell, and then it will be attached by the ribosome, okay, to produce the protein. Okay, that's the process. You see this over here inside the cell, okay, there is a production of RNA from DNA, and this production using special enzyme is called the transcriptase. That's the reason why this process is called the transcription. Transcription of RNA from DNA using this transcriptase. Now, this is what very unique with this HIV virus. They carry the reverse transcriptase. It means that reverse the process. Instead of from DNA into RNA, the process is actually from RNA, okay, into DNA. Okay, of course, the enzyme will be reverse enzyme. The enzyme is called the reverse transcriptase. Okay, So this is the process that is done by these viruses. Is able to make their own DNA from their own RNA. And this DNA then will enter into the cell and will combine with the human DNA. In this case, it is inside the T helper cell or CD4 cell. So this is the integrations of virus DNA in or with the human DNA, and it will stay here forever. And then this part of this gene, which is the virus gene, will produce the DNA, RNA over here, right? Using the transcriptase enzyme that is available in human cell. Okay, so they're using the transcriptase enzyme of human cell. They carry their own reverse transcriptase because human cell do not have this enzyme. Okay, so by using the human transcriptase enzyme, they are able to produce their RNA from this part of their DNA that is integrated with the human DNA. And then this RNA will be used, to make the proteins, like the capsid and also the spike, and also used for making or replicating their own RNA. Okay, so this is a synthesis over here, and then this is going to be assembly. Okay, and then what happens? After assembly, the virus will be released. Okay, maybe 1,000 of them will be released from one infected cell. And then this virus that is released will be available to infect other cell, or because it is located in the blood, will be carried by the blood and then can be transmitted to other people. Okay, so this is the cycle of HIV virus inside the CD4 or T helper of human cell. Okay. Okay, now the effect of this HIV in human body. Okay, the primary effects of HIV infection, okay, will be extreme leukopenia. What is leukopenia mean? Leuko mean white blood cell. Penia mean lower. So it's causing the white blood cell, especially the T cell, which is part of the lymphocyte, to be very, very low in number. And because they destroy this lymphocyte, this T cell, okay, the T helper. And then also can cause formation of giant. And they can also infect the macrophages that release virus into the CNS nervous system with the toxic effect and some inflammation. So this is the primary effects of HIV. And the secondary effects, this is the one that actually causing that patient to die, okay, because of the distraction of CD4 will cause this patient to be unable to protect against some opportunistic infection. Okay, like just regular flu, okay, or regular diarrhea, for example, it can kill this AIDS patient. Okay, they unable to really protect themselves against very mild infection. Okay, usually for normal people it will not cause problem. Okay, now look at these stages of infection over here. Okay, so we're gonna look at this. Okay, this is the number of T helper that will be going down during the infection. Boom, going down, 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 down. Here, over here, this is the threshold over here, which is 200 okay, cell per microliter of blood. So whenever the T helper or CD4 lower than 200 cell per microliter blood, then this person will start to have what we call the AIDS. So the AIDS is starting when this person has a very low number of T helper. Then how long to take this from from the first infection until this AIDS disease? It depends on that person. Okay, sometimes some people maybe only month. Sometimes some people take about 2 until 15 years. Okay, to become AIDS. Okay, from HIV positive, this one until AIDS sometimes take time about 2 until 15 years. It means that during this time, this patient actually do not have really uh, severe symptom, or maybe no sign at all, but they able to transmit or transfer the disease to other from for this long year. Eh? So you can imagine sometimes we don't know someone have HIV positive and they do not have the sign and symptom yet because what? Because they are not in the AIDS condition yet. Okay, but they able to transfer the disease to others. Okay, again, most of the time, this one will have the asymptomatic, which is no sign and symptom for several years. And then after the T helper or CD4 reach 200 cell per microliter blood or lower than that, then they start to have, the patient will start to have the AIDS sign and symptom, which is their immune system will be very, very low. Okay, the testing for HIV, mostly using ELISA tests to confirm the HIV positive infection. Now, the AIDS, when that person is considered as having AIDS, of course, the first measurement is when their CD4, okay, or T helper is lower than 200 cell, okay, per microliter blood. So this is the first measurement. And then after that, there are also several indication of AIDS. Okay, of course, this is the first indication over here, which is here. Okay, CDC provided a list of AIDS defining illnesses. So usually people with AIDS will have this type of disease or illnesses, which is a lot. Therefore, the CDC put it in long list. There is a list of defining illnesses that give the sign and symptom of someone that have AIDS. Okay. So in the skin and mucosal membranes, they have this cytomegaloviruses uh, around the eyes, which is retinitis, which can cause loss of vision. And again, on the skin, they will have herpes simplex. Okay? And then also, as I mentioned, is the Kaposi sarcoma. So these are the list of defining illnesses for AIDS in the skin. How about nervous system? So they will have this type of disease, cryptococcosis, which is, these infections causing some extrapulmonary infection, and also HIV encephalopathy, which is damage in the brain, lymphoma, primary in the brain, and leukoencephalopathy, and sometimes toxoplasmosis of the brain. So it's not only in the skin, the disease, some infection also attack the brain system or the nervous system. Okay, how about the cardiovascular system and lymphatic system? Okay, coccidiomycosis, which is disease caused by fungi. So fungi will enter into the cardiovascular system and the lymphatic system, causing this fungal infection. Cytomegaloviruses, which is the human herpes virus number 5, it can also infect patient with AIDS. Histoplasmosis, this is fungi infection. Okay. Burkitt lymphoma, this is EBV, Epstein-Barr viruses. Lymphoma, Mycobacterium kansasi, Mycobacterium tuberculosis, so TB. Salmonella septicemia. So this organism or this germ, whether bacteria, fungi, or viruses, they can enter into the AIDS patient and causing these diseases. List of disease, in the cardiovascular and lymphatic system. This is respiratory tract infection. So candidiasis, which is caused by fungi. Herpes simplex bronchitis, this is caused by viruses. Mycobacterium, this is bacteria. Tuberculosis, this is also bacteria. Pneumocystis jirovecii, this is caused by fungi. Pneumonia. And, this is all the list of disease that telling us if the patient of AIDS, they usually have these many disease on their body. Because what? Because they are unable to really protect themselves against many infection. GI tract infection, candidiasis, esophagus, herpes simplex, isosporiasis, genitourinary infection, cervical carcinoma, herpes simplex chronic ulcer, and many more. Okay, so this is just telling us how bad is patient with AIDS. Actually, they really unable to protect themselves against diseases because they unable to give the protection from the T helper. So T helper is one of the main white blood cell that protect us against many diseases. And so far, there is no vaccine for HIV or for AIDS. And there is also no cure. So whenever someone have HIV positive, and if they do not really take the drugs or medicine, and then they will go to the AIDS condition. So how about the drug? The drug is usually just used to slow down the progress from HIV positive into AIDS. So the drug that many people with HIV positive take is usually for slowing down the process of HIV positive into AIDS disease. It's not really cure the HIV positive. Just remember, because the DNA of the virus actually stay in the patient cell or DNA. Therefore, it will not be removed. It cannot be removed. The process is only kind of slowing down this. The drug is usually only slowing down the process from positive into AIDS. Like for example, there are some drugs that inhibit the enzyme So if that patient, for example, HIV positive patient take this drug, then the process of making DNA will be slower. But when that patient do not take it, for example, stop taking that drug, then the process will be faster again. Inhibit the fusions of the virus, inhibit viral integrations of virus into the cell, uh, antiretroviral, which is also used for slowing down the enzyme. So all of this drug is actually mostly to slow down the process. And remember, when the patient, HIV positive patient, stop taking the drugs, then the process will be faster again. Okay, so you might heard some people, like athletes or maybe, some artists, they have HIV positive, but they still in HIV positive, for a long, long time. They are not become AIDS patient yet because what? Because mostly they taking the drugs to slow down this process, and the drug price is very, very expensive for this HIV positive. And also the drug mostly will cause the side effects to the person that take this drug. Okay, they might be causing nausea, vomitings, and some other side effects. There is another retrovirus cell, virus, that infect human, is called the human T cell lymphotropic viruses. This is causing some leukemia to some patient. It's a chronic type of disease. Usually the person that have this disease will have some sign and symptom like bruisings, bleeding, paleness in the skin, fatigue, okay, and recurring minor infection. This is less severe compared to the AIDS. Okay, so the leukemia that caused by the human T cell is considered as less severe compared to the other retrovirus, which is HIV, that causing the disease called AIDS. Okay, so again, human T cells viruses, there are two types. Okay, HTLV1 and HTLV number 2. The HTLV number 2 even less severe because mostly the patient will not have the sign or symptom. This is kind of a symptomatic type of infection. Okay, I think that's all for the retroviruses, which is HIV and human HTLV viruses. And again, the most severe one from these retroviruses is the HIV, human immunodeficiency viruses that can cause AIDS, acquired immunodeficiency syndrome. I think we'll stop here, and then the next lecture will be discussing the non-envelope RNA viruses. Goodbye now. Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 25, lecture 4. And today we will discuss the non-envelope RNA viruses. Okay, remember again this classification. RNA virus that infect human is considered as animal virus. Mostly they will have envelope, okay? And because RNA, so mostly they will only one strand of RNA. So it's going to be single-stranded RNA will be the most common type. Okay, therefore, these are the most common type of virus that infects human, which is enveloped with single-stranded RNA. Maybe segmented RNA or non-segmented, or the special virus that carry their own enzyme. It's called the retroviruses. So we already done with this envelope single-stranded RNA viruses. Now we go to the least common one, which is the non-envelope. Non-envelope. Then from this non-envelope or naked viruses, the single-stranded will be most common because this is RNA. And the double-stranded, this is kind of weird viruses. Is very, very rare. The virus that carry double-stranded RNA because mostly RNA are single-stranded. Okay, for the naked single-stranded RNA, there are two groups, okay, picornaviruses and caliciviruses, while for the naked double-stranded RNA will be only one group, which is rioviruses. Okay, we start with the first one, which is the picornaviruses. Okay, pico actually mean very, very small. Okay, so this is after or lower than nano. We have meter, millimeter, then gonna be micro, and then nano, and this is pico, which is very, very small virus. So this is considered as the smallest type of viruses. Okay? There are several types, okay? Enteroviruses, including over here is actually the one that causing the polio, okay, poliomyelitis, and also hepatitis A viruses. It is in the group of picornaviruses, subgroup of enteroviruses. Entero mean the virus that actually go to our GI system, okay, GI tract. Entero mean intestine. And then rhinoviruses and cardioviruses that maybe infect heart and the brain. Okay, again, maybe the most common one is this virus over here, which is polioviruses that causing the poliomyelitis. And this poliomyelitis actually, litis is actually the disease that almost eradicated, but there are still some people have polio, but the vaccination actually able to reduce the number of polio patients worldwide. Okay, this is considered as the disease that almost eradicated. It's not eradicated yet. Okay, as we know, most of the kids right now, they do not have polio compared to the kids on the old time when many people actually have polio. Okay, that's maybe the first, the most common type of picornaviruses, which is causing the polio. The second one may be this one, okay, Hep A, okay, hepatitis A viruses. This is also enteroviruses. It's usually transferred through the food or drink that is contaminated with the viruses. Rhinovirus is usually causing a very mild, common type of cold. Okay, cardioviruses can also causing the and myocarditis, which is inflammation of the brain and inflammation of the heart. Aptoviruses, this is foot and mouth disease. Okay, polio, this is one of the most common type of picornaviruses, which is causing polio or poliomyelitis. And polio, this is a kind of acute enteroviral. So this is enteroviral, is mean that the virus actually enter into our system through the food or the drink, go to the GI tract. Mostly will not cause infection to some people, but about maybe 2% can cause the infection. Okay? So as I mentioned before, eradication of this disease is actually expected, okay, because so many people now have the polio vaccination. It is expected that on the next generation, there will be no people with the polio. Maybe right now you still have some older people that have polio, but mostly kids now, they do not have polio because it almost eradicated. Again, this is the epidemiology or the transmission of polio, mostly through the oral, right? Whether drink or food. Okay, so transmission is fecal oral route. Mostly very mild or even non-symptomatic or non-specific symptom. Fever, headache, nausea, sore throat, myalgia. Okay. Now the problem is if that virus actually enter into the nervous system, especially the motor neuron, it actually will destroy the motor neuron. Motor neuron is the neurons that order our effector to do something, to perform something. One example of effector is our muscle. Okay? So when the motor neuron is affected, then what happened? The muscle will be also affected. Okay? The muscle will not contract, will not move. Especially for this virus, they infect the lower motor neuron, which is the motor neuron that order contractions of muscles in our legs or our foot. And what happened? Causing the flaccid paralysis. So people that have polio, they usually unable to move their legs. And then if they unable to move their legs for a long time, what happened? The muscle become very, very small. And then you see their legs also become very small because of this infection. So that's actually the problem with the polio infection. 90% usually asymptomatic, do not really give the sign and symptom infection. It's just in the stomach, in the in the GI tract system. Sometimes it's just go and then flush it into the fecal. But whenever this virus enter the neuron, especially motor neuron, then it can cause the flaccid paralysis, okay, unable to move the muscle. So this is example of a person with the polio. If you see this muscle on the lower extremities, the leg is become smaller and smaller. When this kid or this girl, this kid growing, okay, their body will be growing, but these legs will keep small, okay, because what? Because the muscle unable to move, causing the muscle to be reduced in strength and also in size. So it stay smaller. So this is the polio. When this polio actually attack the motor neurons, okay, that control the movement of muscle on the lower extremities, on the leg. Okay, treatments and prevention. Again, the vaccine is available. It has been used for several years. And again, it is expected that the polio will be eradicated worldwide. So it means that The new generation mostly will not get the polio disease because of this vaccination. There are some other enteroviruses that will not cause the polio. This is non-polio enteroviruses. Okay, so this is a type of common complications of the enteroviruses, common cold syndromes, skin rashes, hand, foot, mouth disease. And again, this is mostly the non-polio enteroviruses. Okay. They also can infect the around the eyes. Another subgroup of pico or picornaviruses is the one that causing the hepatitis A, Hep A disease or virus. And again, this is transmitted through the oral transmission, which is through the food or water contaminated with the viruses, the picornaviruses. The next one, the one that do not have the envelope, okay, naked virus with the single-stranded RNA is the rhinoviruses. Okay, usually associated with the kind of giving a common cold sign and symptom. Okay, so this is symptom of someone that has contamination or infection with these human rhinoviruses: headache, chills, fatigue, sore throat, cough, nasal drainage. And mostly it's from oral transmission, so food and maybe drink, or maybe the hand, or the one that hand that may be infected or have some virus in it, it can enter through the mouth. The next one is caliciviruses. This is also the naked, non-segmented SS RNA, so single-stranded RNA is called the caliciviruses. Again, mostly fecal oral route, so the problem mostly in the GI tract, so causing the nausea, vomiting, cramp, diarrhea, and chills, because this is a GI tract infection. And the name of disease is called the Norwalk disease or Norwalk agents. Norwalk is actually the name of area or small city in Ohio. So this is the first case of this caliciviruses. It's called the Norwalk enteritis, which is Norwalk gastroenteritis, is inflammation of the intestine. Okay. The last one, and this is the most or the least common one, the most rare one is a double-stranded RNA over here. It's only one, which is reoviruses. Okay, so naked, segmented, double-stranded RNA. Only one virus is called the reoviruses. Mostly is enteritis, especially on baby. Yes, this is the infections mostly on baby, okay, infant and children over here. And mostly this is enteritis, which is like a diarrhea, okay, type of disease in the GI tract or GI system. Okay, the next one it is prions. Remember, prion is not virus. It is just a protein. Okay, protein or misfolded protein that can cause infection to human. Mostly, okay, the infection is in the brain. Therefore, it's called the encephalopathies, which is TSE. Okay? Spongiform, uh, transmissible spongiform encephalitis or TSE. Okay? So mostly on the brain. Example in human is called the CJD, okay, which is the Creutzfeldt-Jakob disease. So this is the most common type of TSE in human, okay, Creutzfeldt-Jakob disease. This is in brain damage. It's causing brain damage. In animal, it can cause what we call the cow disease, mad cow disease. So we can put mad cow disease over here. That's prions. Again, mostly in human causing the spongiform encephalopathies. It means that the brain cell is become like a sponge. It's destroy the brain cell because of this infection. Mostly the prions actually transmitted through the food, especially if someone eat the animal brain. The animal brain that maybe have these prions. Like for example, mad cow disease, the mad cow, that cow okay will have the prions on their brain. If people eat this animal brain, then that person might infected by these prions, causing the TSE. Again, human TSE, transmissible spongiform encephalopathy. Mostly, the most common one is the Creutzfeldt-Jakob disease or CJD. And again, this is a type of spongiform encephalopathy, which is the, if you see this, compare the normal cell and the normal brain with the TSE brain. So you see the brain is kind of, the brain cell is kind of broken down over here. It's not that nice, compact like the normal brain over here. Okay, so it's causing the damage in the brain cell. Okay, again, just comparing the normal cell with the cell of person with TSE. Okay, the preventions, the main prevention is avoidance. That's the main prevention, avoidance to eat or to have contact with the contaminated tissue, mostly brain over here. Okay, animal brain. So try to avoid eating the animal brains, especially if that brain, they have the prions on it. So no treatment actually available for this type of disease. So that's the only way to prevent against disease is to avoid eating the animal brain, especially cow brain, mad cow brain. I think that's enough for the naked RNA viruses. And then we will meet again on the last lecture, which is special discussion on arboviruses. Goodbye now. Hi, everyone. Welcome to Medical Microbiology. So today, going to be the last lecture of our chapter 25. And this last lecture, we will discuss especially about arboviruses. Now, what is arboviruses? This is actually a group of virus that is transmitted by vectors. Okay? So vectors are animals that carry disease. So these arboviruses, could be DNA viruses or RNA viruses. Okay, so any virus that is transmitted via vectors or carried by vector, especially that vector is called the arthropod. Okay, so arthropod is animal that have their legs coming from the chest. So this is arthropod, this is chest. Arthro mean chest, pod mean leg. So animal that have leg on their chest, like for example insect, the mosquito, the tick, flies, gnats, they all considered as arthropod. So this group of animal can carry virus, okay, from maybe animal to human, or maybe from human to another human. Okay? Therefore, any virus that carried by this arthropod is called the arboviruses. So arthropod, okay, arthropod-borne viruses. Okay, therefore it's called the arboviruses. Okay, the virus that carry by arthropod vector, by mosquito, ticks, fly, gnats, for example. Okay? There are about 400 viruses that carry by this vector, by the arthropod vectors, and mostly in the tropical countries. Why? Because many arthropods like mosquito, tick, flies, gnats is actually, uh, you know, uh, available or live in the tropical areas. Okay, therefore, sometimes the way for removing or prevention of disease that causing by virus that carry by this arthropod is by uh, distractions of the arthropod itself. For example, using the insecticide, for example, to kill the or to destroy the mosquitoes. Okay, so when the mosquitoes kill, for example, then the transmission of disease that caused by this virus will be lower. Some virus that can be carried by this arthropod, like toga viruses. This is one of them is the one that causing the rabies. Okay, fluffy viruses. This is the one that causing the dengue fever. Okay? Zika virus is also in the fluffy type of viruses, and Hep C type of viruses. Okay? Bunia viruses, this is actually DNA viruses, and Rio viruses. Right? So this is some of viruses that can be carried by arthropod. Okay? Disease that causing by this virus may be mild, okay, mild fever, and sometimes it can cause life-threatening, like for example, fluffy viruses over here causing the dengue fever, which is hemorrhagic fever, especially in the tropical country. It's actually life-threatening hemorrhagic fever. Okay, when the patient infected by these viruses, the fluffy viruses, causing this dengue fever, and then what happened? The patient might be die, and then they actually have bleedings, okay, from their body, okay, from their nose or maybe from their skin, the pores of the skin. That's fluffy virus that causing dengue fever is actually carried by mosquitoes. Okay, one of the arthropod vector. Okay. Again, the factor of these viruses tend to be clustered in the tropical area, okay, tropical country or subtropic country. So many tropical country or subtropical country, they have a lot of infection or viral infection because what? Because they have the factors that carry or transmit the disease from animal to human or human to another humans. Okay? And again, the arbovirus life cycle will be closely tied to the life cycle and the ecology of the factor itself, because most of the virus actually will be carried inside the factors, part of the cycle of the factors itself. Okay, and as I mentioned before, again, controlling the factor, like using insecticide, for example, can also control the spreading of disease. This is the distributions of arboviruses, and again, mostly in this, you see this one, this is a tropical area, tropical or subtropical area. In this United States, mostly on the southern part, where is the temperature over here is also kind of tropical temperature or subtropical temperature. This is the spreading of the arboviruses. The virus that carry by the arthropod factors. Okay, many of these viruses can cause what we call the viral encephalitis, which is brain damage. Okay. Example in the United States is like a West Nile virus encephalitis, and again this is carried by mosquito. It's come to the United States. Eastern equine encephalitis. This is the Eastern United States and Canada. Lacrosse encephalitis, mostly in the Appalachian Mountain area, Midwest of the U.S. St. Louis. This is the mid area of United States. Chikungunya viruses, this is mostly on the Caribbean area. Zika viruses, this is also in the southern of America. Sometimes it's also enter into the United States when the mosquito or the insect or the arthropod factor that carry this disease also available in some area in the United States. Yellow fever, it has been eliminated in the United States, and this is one of disease that also carry by the arthropod factor, and this is the one that causing what we call the hemorrhagic fever. Hemorrhagic fever mean the patient will have higher body temperature with type of bleeding. Sometimes they will bleed through their nose or maybe ears. Okay, that's the hemorrhagic fever. I mentioned before about the dengue fever over here, mostly in the tropical area, and this disease is carried by special mosquito. It's called the Aedes mosquito. So when we control this mosquito, it will also control the spread of the dengue fever. And dengue fever causing what they call the hemorrhagic shock syndrome, break bone fever, when they have a extreme muscle and joint pain, and it can be fatal, especially for kids. Okay, so kids, if they have this dengue fever, mostly it can kill them within 4 to 10 days. So they need help as soon as possible. Okay, I think that's all for this chapter. So we finish chapter 25, And also it is considered as a finish. We finish module 1 of this course. So module 1 finish, then you will have, the exam for this chapter, which is exam number 2, okay, for chapter 4, 5, 6, 24, and 25. Okay, so be ready and good luck with your exam number 2. Goodbye now.